Uh, so, good afternoon, everyone, and welcome to this webinar, international webinar on COVID-19 pandemic, hosted by Institute of Bioinformatics. And basically, Institute of Bioinformatics is a non-profitable organization which was started by Professor Dr. Akhilesh Pandey, and who is currently at Mayo Clinic as well. Previously, he was at Johns Hopkins, and he started just after Human Genome was published, or was in the process of publishing. He realized the importance of genomics and bioinformatics and translational research. So at that point, he had started this organization, and noticeable work was the human proteome data and subsequently several other publications. So you can follow on our website, and we have already explained you. In third day of this seminar today, we have 6 speakers. First, Raja will speak about NGS based applications for SARS-CoV vaccine development, and then Gaurav Sharma will talk about evolution and diversity of coronaviruses, and then Dr. Tikam will be speaking on peptide based therapy, and then I will discuss about understanding of nucleic acid proteins and vaccines for SARS-CoV-2. And then our eminent guest from Israel, Dr. Professor Malik Yusuf, will be giving insight from his work on machine learning. And finally, we'll have Mitin Jain, who is scientist in University of California, Santa Cruz, and he's a pioneer in nanopore sequencing. And if you are following what is going on nanopore, in last 5 years, several major publication, he is the lead author. So that will be our immense pleasure to learn what is going on nanopore sequencing side, and I assume Raja will also discuss some part of it. So let's begin with Raja's talk. Dr. Raja is the CEO and founder of Genotypic Limited, Bangalore, and he was in his education was in Madurai Kamaraj University, and subsequently he has postdoc from abroad, notably from Israel. And after that, he came and he opened one of the genomics company in India, and his company is expanding. As we know that he has probably 2 or 3 sister companies, and it's fortunate for me and my team to host him here, and lot of potential of collaboration and for others as well. If you have a sequencing facility or so, funds or so, do consider contacting Raja. And Raja, stage is yours. I will stop my presentation. Okay, Raja is presenting, so I will just stop mine. Okay. So, Dr. Raja, please go ahead. Yeah. Raja, can you hear me? I have to stop my presentation. Yeah. So, oh, Dr. Raja, can you guys confirm me whether you are hearing me? Dr. Raja could not hear. And let me introduce Dr. Raja again then. Raja, please confirm me if you are able to hear.
See, I'm speaking from another phone. Can you hear this?
Yeah, I can. We can hear that.
Okay, so maybe I should use the other
So
line, but you can see the presentation, right?
Yeah, sure.
Okay, so let me start. So there is no video, right?
Video is there. I am able to see your slides, and we are on live on the YouTube.
Okay. So there is no, the camera is not working, only the presentation, right? The voice.
Yeah. Camera is not working from your side.
Okay, it can work, but I don't know why it's not working.
Yeah, you are in Mac, I guess, and Mac requires permission. It's a competitors software we are using, right? So
no, I am actually using
okay.
Regular PC only.
Okay. So then they're also setting
so
my.
So how do I?
So normally
there is.
Normally there is a in your Google itself there is option. We do not have the control over your because.
Wait a minute. Let me see in the
yeah
Google Doc. So if I can go there and see presentation audio is there.
Yeah, presentation video is not.
Doesn't allow anything else.
Yeah, so we all are agreeing that Google has just come up without full. In my system, I can see both actually audio and video option both.
Okay, so here it's not there. Even in my mobile phone, it's not showing up.
You see your CEO. Okay, so basically for CEOs they have special safety measures. So yeah, because this is not in full okay. Developmental stage itself. They have released it, I guess. To complete.
Fine. So should I start?
Yeah, sure.
Okay, so let me start. Okay, folks, it's a great day today. Thank you, all of you, particularly the folks from Institute of Bioinformatics, Abhishek, Munish, and all of you for giving me this opportunity. So this is all about SARS virus and what it is doing, what we can do, when is the vaccine coming? You might have a lot of questions. So let me proceed. So as you all know, Genotypic is the India's first genomics company. We started providing what is called as the do-it-yourself products. That is, you can take a nanopore sequencer and sequence everything yourself. So this is what currently we are doing. Started in 1998. And then now we are in the COVID era 2020. So what's going on? See, I put my picture itself because, as you know, this is how I look. Since video is not there, I was like this. Now you don't want to see me on this Zoom eras. See, currently my mission is to apply genomics in the COVID-19 diagnostics. In screening, in therapy, vaccine development, everywhere we are applying genomics. So I'm going to tell you how it's going and where things are going on. See, you all know the pathogens are ubiquitous in our environment. It's everywhere. Whether you wear a mask or not, even in the mask, parasites are sitting, right? There is billions and billions of bacteria on our skin, in the environment, in the food we eat. Everywhere there are bacteria and fungi and virus. Everything is there. So, where are these parasites? I guess someone is talking.
Please, Mr. Pratap, can you switch off your sound?
Thank you. So, parasites are everywhere. It could be bacteria, it could be fungi, it could be viruses. Even now, there are lots of viruses everywhere. And of course, our SARS-CoV-2. And this is very similar in the effect of to smallpox. You know, smallpox virus was killing. If you get smallpox, there is a 50% chance that one will die. 50% chance. So you should thank SARS-CoV for not really killing us. The kill rate of SARS-CoV is so low, but we are almost as afraid as we are having smallpox. Smallpox, at least you know, all those little bubbles coming everywhere. SARS-CoV, you don't know what's coming. Only when something hits, people know. That is dangerous, and people are scared not without reason. So what is this SARS-CoV? This is an RNA virus. It is just a 30 kb genome. For an RNA virus, this is slightly larger. You know, HIV and all these viruses are sub 20 kb. So this is, okay, large RNA virus, but a very, very tiny virus. See, all these have become ubiquitous now, right? Somewhere I go, I tell them, hey, boss, please don't fumigate me. People are even spraying something, right? You go to some hotel, someplace, offices, people are spraying. I don't know what the hell they are spraying. But these have all become, see, all of you are wearing mask when you go out, and God knows whether it works or not. But everyone is saying, and if you don't wear a mask, people think you are an idiot, right? And if someone don't wear a mask, you think he's a bad guy. See, nowadays, bank robbers are going without mask. Everyone else is going with mask. Things have changed. So what can SARS-CoV do? It can kill. And it can kill quickly if it does kill. So I just want to show you what it does. See, here is this little red dots. That's the SARS-CoV. Once you get exposed, in less than 1% of the cases, it goes to the what they call as upper respiratory infection. 90% cases, if you are on prolonged exposure, that's in your own homes, few people are having SARS-CoV, the COVID, then you can get exposed. Then what happens is, after this step, 98% of the cases, these are all healthy individuals. Healthy, I mean someone from 20 to 50 years. Or maybe 45, it all depends. So the 98% of these cases, one gets immunity. Let's talk what is this immunity. Whereas in 2% of the cases, it can go to the lungs and it can cause death. This is for a healthy individual. You're not talking about immunocompromised, old people, everything. For healthy individuals. So what is a vaccine? I'm sure you all know what is vaccine. You might have heard this thing, no, attenuated virus. For example, if you take a human virus and passage it in rabbit for hundreds and hundreds of generations, it will maintain everything, but it will lose the ability to infect a human, a serious disease. But that will be a mild infection, right? So this attenuated, this is how all original vaccines were made. Either they were, for smallpox, we used cowpox as a vaccine because they're related to each other. Cowpox can't infect human. Many of the vaccines today are all attenuated vaccines. And people don't know because the sequencing was not there those days. So they know it can make a mild infection, but they know sequence is different. But now we know what are the sequences. Those days people don't know. So now what happens, 100% of the cases can develop immunity. See, I'm saying 100%. Maybe it's 99%, 99.9%. But you all heard of this herd immunity, no? If most people are immune, there is no one to actually pass that virus to you, even if you are not immune. So for a vaccine, it need not be 100% of the people who take the vaccine should develop immunity. Even if 80%, 90% is okay, basically. At least human race won't die, and then so-called herd immunity can come. So as you know, right, there are T cells and B cells, right, memory cells. That's for cell-mediated and antibody-based immunities will arrive. So what is the SARS-CoV genome? Let's go to the SARS-CoV genome. It's 1 in 1,000, 1 in 100 million of a human genome. And you see, it can do so many things. So let me skip these slides. See, the COVID epidemic is completely different. Even before it became pandemic, epidemic is a region like in China, Wuhan, it started. See, you can see I have even made one side is genotypic logo, another side is COVID. Now this is Karnataka politics. Whoever is in the forming the government, you join them. Take their logo, join them. That's the idea. So what's happening here? In January itself, 200 millions went to that sequencer. See, I'm having the sequencer with me. Unfortunately, the video is not working. If it works, I will show you. Maybe after I close the presentation, I think the video will come. That's what I think. So you can see there is huge number of sequences are happening. They sequenced and then they found it's a SARS virus and what are the sequence differences. Everything is known in January itself. So it's interesting, right? So where is genomics used in vaccine development? First, even at the start point, you rapidly sequence the outbreaks. In Wuhan, it was sequenced. It's sequenced in India. We ourselves have sequenced more than 100. Thousands are being sequenced in India, right? And I'm going clockwise. So you sequence the epidemic strain and you sequence the pandemic lines. That's in India, there is line. Now you know there is some 12 different clades, 6 major clades there shortly. Sometimes some people are naturally resistant. We also see even smallpox, people know some people were resistant to smallpox, right? And some people in Africa are resistant to malaria, the ones who have a mutation in hemoglobin, right? You know that. So like that, SARS also some people are resistant, but we don't know who are resistant, right? And then you also want to know the pathogen genome, find the proteins, then you can find what are the ones which can be used as a vaccine. You need not have the whole virus, maybe regions of the virus which can be a vaccine. You can also deep sequence vaccine batches. This after the vaccine is made. Got you guys? After the vaccine is made, you have to sequence the vaccine batches to know it is, I'm jumping on this side, it is indeed attenuated. Okay. So you can also express the genes, right? You can also look at cross reaction. Some people are looking, is there any other virus, any other vaccine we know which can cross protect? Unfortunately, there is none. That's a problem. And then you also have other agents for quality control. Everything can be done. So this is slightly old to see how many. See, now there are 8,000 genomes sequenced. This was only a month ago. You can see approximately 2,000 genomes sequenced. We are using the nanopore. India also, you can see 120 genomes sequenced in India, right? So how is this sequencing upward? Firstly, we have to choose the vaccine strain. Until then, you need not. So to make an attenuated vaccine, you need to have a vaccine strain. So that's where we are. So, and then this is epidemiological research. where is it spreading, from where it is spreading. So is there any mutation? If you can reduce the virulence, you can actually have a vaccine strain, something which spreads well, but it is not causing a serious disease, is an ideal vaccine strain. Right? So these are all the global databases. You can go to this website, GISAID, if you put, you will see there. You can go to the site and then you will know ID, if you put, you will see there. You can go to the site and then you will know from where and how this is spreading. It's made of 6 major subtypes. See, this is the first outbreak and everything is an outcome of this first outbreak itself. So there is nothing like influenza virus. No, you have different outbreaks and all. Maybe the second round might be a different outbreak. We don't know. So let's go ahead and see it, what's happening. So these are the 6 major subtypes. And you might know what is called as widely shared polymorphisms, WSPs. So here are the major polymorphisms, and people have already known amongst these 12 types, and then you can see each one, how many of them are changing what amino acid. See, this is very important because once a vaccine is made, a vaccine which is effective against one, the wild type, the first one, may not be for certain variants because the antigenic sites are changed. But if you have a polyclonal vaccine, antibodies, or the whole viral vaccine live attenuated, this may not be a big problem. But in influenza, this is indeed a big problem. So it's important. It's very important. So what do we do with the genomics of the host is also important. People many times forget it, that, see, we all already know certain vaccines don't work for Caucasians, it works for us. PCG is one such example, I guess. So the genomics of the host also has to be studied. But we don't have that kind of a time. Now we are spraying something. I think some people are even spraying petrol and lighting themselves up. It's worse situation now. See, one reason you have to sequence is to understand the biology of the virus, right? And use the outer coat, what is inner coat. So that's where you're going to develop the vaccine for, right? And this is on the host responses, maybe certain HLA, right? Certain IgG locus may not even need a vaccine. See, already people know if you are blood group O positive, I think they are almost resistant. The jetting is 0.1% in comparison to, I think, A. A negative is more susceptible. This, I don't think they have done it in India. It's data coming from Italy and China. So how do you attenuate a virus? One is, as I told previously, infect into rabbit or maybe mouse cell lines, you passage many times. Then you get an anti-virulent virus. This can make a mild disease, but it will give you immunity, but it will not kill you. Another thing is, use genetic engineering, knock down certain amino acids, certain regions to make it an attenuated virus. But this is a dangerous game. If you knock it down at multiple places, that's a good way to do. So there are multiple types of vaccines. That's something one had to say. First is the particulate vaccine, DNA vaccine. DNA vaccine is you put some of the genes of the virus, put it into a piece of DNA, and then you inject it into your muscle cells with a human promoter and transcription by translation initiation sites. So the muscle cells will start expressing the viral proteins. For quite a few diseases, they have done it, but it's still not a very, very highly prevalent method, but that's the fastest way to develop a vaccine is a DNA vaccine or a mRNA vaccine. Right? All of them have been tried. There is heat inactivated whole virus. This is the most simplest way. Just grow the virus in the lab, heat kill it, inject to people. But the problem there is you need heavy loads. See, an attenuated virus is the best vaccine because it will divide for yourself and develop substantial amount of immunity. And then this recombinant DNA virus vaccines, that is clone it into adenovirus, small pieces of this, antigenic ones, which can raise antibody. That's another way. So where it is used, here is a summary. In the human side, this is the host side. You can look at the genome, epigenetics, right? Age, why upon age, apart from immunity, what else changes someone's susceptibility? That also we can see. So this is a summary of whatever I've spoken so far. See, a vaccine also changes the microbiota. Like BCG vaccine is known, one of the cases. It's well known that microbiota. So whether people are now even saying, maybe I will do BCG and I will get immunity to SARS-CoV-2. They are nowhere connected. BCG is a bacterial. This cross reaction is not by sequence or antigen similarity, but in general induction of immune response. See, there are lots of sequencing platforms I've given. I'll send this to Abhishek or Munish or someone in IOB. You can get it if you're interested. So I'm coming back. I think you have learned all these possibilities. See, attenuation, no one is doing now. This people are doing. This people are doing. This people are done already. That is how, from sequence data, people found this O blood group, by the way. This, not many people are doing. This, if such a vaccine comes, people will do. Let's hope so. These people are extensively doing NGS. This people did and found it's of no use. This people are going to do once the vaccines. Just for people who have never seen a nanopore data or a sequence data. See, here is the first 5 reads, which is nothing but in nanopore, it is just electric signal. See, nanopore sequencing is nothing but looking at the shadow of the DNA in an electric field or RNA in an electric field. This is like our Taleva, Rajinikanth, no? He see your shadow and tell you who you are. Whether you wear a mask or not, he can find you. So that is this. So that image file or the wave file is converted to FASTQ, nothing but FASTA with quality scores. The Q is quality. So you can see the sequence, and here is the quality for every base, every position there is a quality. And then finally what you get is the FASTA reads, which I think you all know what to do with it. So what else is genotypic doing? Finally, I'm going to give you the verdict. Next slide. So anyone who's sleeping or switched on and doing something, please come back. I'm going to show the most important screen, next screen. So what is genotypic doing? We have made some 100 primer pads, optimized it. We are selling those pools of primers. This comes from Arctic. It's a public domain sequences, but we have re-optimized them. And we are giving everything. Some are from genotypic, some are from elsewhere. So you can do the sequencing yourself. I hope the video will come and I will be showing you the nanopore sequencer shortly. So guys, this is very important. Do you want a vaccine or our coronal? think about it. As of today, the choice is not yours. Maybe coronal is your only choice. There is no one else is even saying there is something which you can use it. So thank you guys. Let me go back and stop the presentation. Maybe I can see you all.
Thank you, Raja. Thank you for a nice, yeah, we can see you. So,
Yeah.
let's have some questions, and if you have a question, please let us see. The questionnaire question will be on the side, and so
Yeah.
super presentation, no question raised. Okay, explains the difference between coronal and vaccine. Tough question.
See, 1 minute. Let me just go out and come back.
Yeah, sure. So
Yeah,
basically,
but you can ask the question. I'm in the other line, so
yeah.
I should be able to see. Yeah.
So explain the difference between coronal and vaccine, and then
That's a good question. See, the coronal is just a some herbal cocktail like neem juice. Neem juice will be sweet, right now, guys?
yeah.
So neem juice, something he would have mixed it up, maybe read some old poetry and mixed it up, and then he says, "This is coronal. Please eat this tablet." Okay. See, almost everyone is doing it. ITC, Unilever, you take many companies, large companies, small companies, all of them are providing something, right? It's 100% protecting, 99% protection. See, no one guarantees it, right? So he is also doing, see, the vaccine is the only way. Vaccine is the only way for protecting someone, but that is far away. So the only chance is, in Tamil we used to say, Aduva poiru. That's a fact. This virus has to go by itself. Because it's infecting a host, it's moving from one person to another person. I think it's good for virus not to kill anybody, right? So the virus might slowly fade away from a deadly virus to a innocuous virus which will not cause any disease. This is something that might happen. But a vaccine is a vaccine. That's the best way to deal with anything. Is there any other question? I thought
There will be. What is
I think maybe
your?
I will read the question here itself.
Yeah.
See,
Sure.
someone is asking why attenuation strategy is not getting implemented. I don't know. Because see, attenuation usually takes a lot of time. Lot of clinical studies has to be done to prove that this strain will cause a mild infection and this strain is not going to kill someone. So this is going to take a longer time. So a kind of attenuation today, what people are doing is take the genes of coronavirus, put it in an adenoviral vector, which is a DNA virus also sometimes. Put it in an RNA or DNA virus, only small pieces. I don't know, maybe it will create a new virus which will clean up the whole mankind and all our problems are gone. But because of the inherent dangers maybe, and also the time taken, maybe that's why people are not using any attenuation as a strategy. Instead, people are growing it, killing it, and making it. So someone is asking, a study shows viral antibodies are seen in infected patients for 2 to 3 months only. So how will the virus? See, very important is, if you know immunology, right? When something goes inside you, it creates what you call as initial immune response is IgM. Then when it's reinfect or the load becomes more, then you get what is called IgG. Then once the virus is cleaned up, then the antibody levels will go down, because whenever you have antibody means you are having the antigen or you had the antigen in the recent past. So after 2 to 3 months time, you will not find any antibody. That is the way, because otherwise we will have antibody against everything. Our whole blood will be only antibody. Only when antigen comes, again the antigen comes, it will increase. So when the SARS-CoV attacks you next time, then the antibody will come. Otherwise the antibody will go down to such a low level, you cannot even detect it. That is the whole idea of immunity. You have memory cells, but not antibody making cells. So 2 to 3 months, you don't have it anymore. So if you don't check it during this season, whether you have antibody or not, after 2 to 3 months, you will not see it, and you will not know whether you are infected or not also. So that's the catch. So COVID-19, see, evening sir, what is the take on unrelated virus interacting with co-infected patients? See, there are many things are going on. I'm not the expert in it. See, I'm a guy who's looking at the genomics of everything, not even this virus. So I'm not the best person to answer co-infection. This is all immunologist or microbiologist, virologist have to answer. Obviously, when you're infected by something, people always say, no, when you have viral fever, you will get a bacterial fever also, because it will injure your throat. Bacteria can enter now. So obviously, if you seriously get infected by virus, that's why sometimes they give antibiotics, because not to save you from virus, to save you from the co-infecting bacteria. So that's where it goes. Next question. What is your take on, so this is done. See, the SARS-CoV-2 is not prone to mutation. See, we sequenced a lot of them. Each one has only 7 or 8 mutations from the wild type. And some of them are because it is that 1A, that clades, no different subtypes. They're all subtype specific. Very rarely we are seeing. See, that is why we have to use this term, the term which I was telling you. See, there are 2 terms there. Basically, let me show. I don't know whether I can share the screen. See, there are something called widely shared polymorphism, that is WSPs. This means this line came from Iran, this line came from China, like that. And then there is also non-widely shared polymorphism. Maybe it's only in Bangalore, only in Coimbatore. Those will be the start of the next strain. So see, the viruses are so small, they don't have scope to mutate too much. That is why you can make a vaccine. But if different blocks and different kinds of viruses are originating out of this, then you will need many viruses, like many vaccines like this, our influenza virus. We have lots and lots of vaccines, types of vaccines, because for every strain. So it's not prone to mutation. It can mutate, but it's not mutating like hell. Someone as Prabhakaran Tyagarajan, Vanakkam Thalaiva, you put your address. Someone is asking the same question. If you found a sequence similarity to another molecule, you mean another molecule is another virus? See, the coronavirus itself, they are all sequence-wise similar, right? There are many similar viruses. people never made a vaccine for them. So they don't know whether it will give a protection. So it looks like in India, people are protected to some extent. Don't trust me. To some extent, people might be protected. I don't know. Someone is saying, "Hi Raja, great talk, Manikaru here. So what is your prediction that when will be a vaccine available? Either RNA vaccine or attenuated viral vaccine." See, attenuated virus vaccine, I don't know anyone is trying. At least the people who are sequencing with us or we know of are not trying. The Chinese will try anything. So they might be trying. Because attenuation itself is a process which takes time. Genetic engineering, we can do the attenuation. See, we can even make a virus from the scratch. But many people are putting pieces of this virus into other viral vectors. And see, some of them, I don't know how I can show you this screen. See, a novel coronavirus landscape. How can I? See, now you will not see me, but you will see the... See, I want to show you folks something. Can you all see the screen?
Yeah.
Is the screen visible, admin folks?
Yeah.
Yes.
Okay. So you can see this first one, the University of Oxford AstraZeneca. This is a non-replicating viral vector, right? So you can go in, see this is what is being used for MERS, influenza, TB, chikungunya, all these things. I think some spelling mistake. All these things are using this one. So this is in phase 3. See, this is what our Dada Garu, no? Dada Garu means Bade Bhaiya from Serum Institute, right? Poonawalla has manufactured some few million doses of this because it's already reached phase 3. And still no one knows whether it will work that well or not, but it is there. He has made it. And Trump will ask him to send everything to US, whether it works or not. That is his idea. He can make his money. So this is the top candidate via vaccine. I think we might be sequencing this. See, many times, no, the vaccine manufacturers also don't want to sequence it. Because if I sequence, I will tell something. No, they don't want to hear it. So you can see here, non-replicating viral vaccine. These are fairly easy to make. See, RNA, right? Inactivated. I was telling, no, inactivated viral vaccines. See, these are all only Chinese guys can do. Because see, in India, we are all scared of even growing the virus in a cell line or something. They'll be growing in fermenters. I think that is how maybe it came out of one such lab. I don't know, I'm just guessing. Okay. Inactive. See, there is protein subunit. These are called subunit vaccines. Okay. This works for some, it don't work for some. So you can see this Nova means probably Novartis or some large companies doing it. Okay. Now so many people are doing, I almost forgot, from Pfizer to even a startup they're doing. See, these are all Chinese academies. See this, anyone says inactivated means the fellow is growing this damn virus. And it's very dangerous to grow that much. See, there are some consortiums. See, there are protein subunit vaccines. Means you make the protein, purify the protein, inject it with the anti-adjuvant. Like BCG itself is in a way an adjuvant. Some people are using spike protein. See, God knows what's happening here, I'll tell you. See, there are 125 candidate vaccines. See, these are all the, what I said, no, DNA vaccines. See, the DNA vaccine has never proved to be so efficient, but it's very easy to start with. In animal studies and all, it will work. See, it will easily pass all this preclinical stuff very easily, okay. See, all DNA, you just do electroporation, okay, or just inject into this one. See, here, some people are asking, no, why live attenuated? See here. Indian immunologicals, but they got it from somewhere else, looks like. This is in Hyderabad. Serum Institute is also doing this. Okay. This is codon de-optimized. That it means it will grow in rabbit, but it will not grow in human, something like that. You de-optimize. That is how natural attenuation happens. Now they have synthesized the RNA. See, in the genomics world, you can do this. See, you can see. See, I think all you folks can go and look at this yourself. There's so many. See, non-replicating viral vector. That's also easy to do, right? Yes, protein. See, this biological E is a Hyderabad-based company. See, next to Chinese, the Hyderabad folks are the ones who have some guts to do all of these things. Rest of India, no. People don't do anything. They will say, what will happen if it goes out and all. So you can see many of them are protein subunit. These are also easy to make. It's just protein purification, right? See, I'm just running it through. So folks, let me come back. I don't know where I left.
Yeah, so basically Raja can, Raja is prepared, and so if you have a question and if you have some industrial inclination, Raja will be a person. Every day we have bring one person who will have entrepreneur and industrial experience. Please raise your question by email, and also not very basic one which we can Google it. So he is a CEO, will not have time. Sometime we have to seek appointment 1 month in advance, and so
I'm jobless, boss. Okay.
so no, sometime you are going to Israel and USA, you London, so this also I'm counting, right? So those months I'm counting. So you are then he's very friendly and available, and but at the same time he has to run the business. So something which you can Google, please Google, and then raise some meaningful question in person to him, and then we will thank him for. You see, he took you to all the vaccine from World Health Organization chart, and so you can also go and check what is going on, and raise some good questions, and hope the other speakers will also be talking about vaccines, and some other you will have chance and also will discuss, and with this we will take on to the next speaker first by thanking Raja. Thank you, Raja, for such a nice talk and discussion. We are very pleased to have you here, and we really thanks for your time and energy, and let's catch up sometime, and please everyone, video will be available on YouTube. If you have missed something, you can go and check, and address and all, he's CEO and Genotypic Bangalore you write, or Genotypic India you write, you will get it. So this is or you'll get in touch with his company. So this is very easy. So and he has a very wonderful company offices and all. So please let's go to the next talk. Thank you, Raja. So our next speaker is next speaker is inside. So our next speaker is Dr. Gaurav Sharma. He is a young faculty at Institute of Bioinformatics and Applied Biotechnology, shortly called IBAB, and he has several years of experience in USA as a postdoc after PhD. And he's primarily microbial bioinformatics and genomic expert, and he has started working on coronaviruses, and I welcome him to give a talk and tell about his group as well if possible, and let me go to the Yes. So, sir, are you able to see the presentation?
I think Gaurav's microphone is off.
Okay,
He
Gaurav's
has to
micro.
switch it on himself.
Yeah. Gaurav.
Gaurav, you have to switch on your microphone.
Okay. Let me also try Gaurav. Just a minute, maybe I have pinned you. So, okay, so the program for today is since Gaurav is setting up, we'll have a run of the program maybe once. Uh, in a window, I will run a program. No. Okay. Okay, so somehow I'm getting a call from. Yeah, hello. हाँ नमस्ते. नहीं अभी आई मीन वेबिनार मैं आपको कॉल करता हूँ. So let me start with just a minute. I will just read out what is the program because it's one of the speaker has asked. So after Raja Gaurav Sharma will speak on evolution and diversity of coronavirus, and then Dr. Tikam will be speaking on peptide based therapy for COVID-19, and then I will speak on the understanding of nucleocapsid protein of SARS-CoV-2 and vaccines in general, and then Professor Dr. Malik Yusuf will speak on machine learning based analysis of vaccine schedules and rates of compliance. It reveals the correlation with COVID-19 mortality, and then last speaker will be Dr. Nitin Jain, who will be coming from University of California, Santa Cruz. So basically, it's a early morning there. So he will be last speaker so that he's awake, and he will be talking about nanopore. So Gaurav is calling. Okay. Hello. I will just give a call to Gaurav so that hello. Maybe we can shift you to next talk, and then by then you can fix it.
No, I'm just trying to restart my system and.
Yeah. Okay. Okay. We just wait for 5 minutes. Yeah.
Sorry, Abhishek.
No problem. It's nothing to worry about. It's all technical glitches will be there. So we are doing for the first time. So don't worry about it.
These things happen like at just a moment.
No, it happens. It happens. So it's also pandemic is teaching us.
By restarting, it will fix.
Yeah. We will just wait for 5 minutes. Yeah. Okay. So as we see that we have kept extra time, so we are aware of such. Okay. Okay. So probably nobody heard. Actually, Gaurav is restarting the computer, and in between I was reading out the the who are the presenters today. So after Gaurav, Dr. Tikam will be speaking, and after Tikam, I will be speaking, and after that, Professor Dr. Yusuf Malik will be speaking. And if Professor Yusuf Malik will want to speak before me, that's also possible. So that he can message me privately. And then last speaker will be Dr. Mitin Jain, who will be talking from California. So there will be early morning in California, around 6:30 or 7, so something like that. It will be 11 hours back from here, so he will be getting up in some time, so we have kept him last speaker. And so Gaurav is restarting the computer. We just wait for him for a couple of minutes. Even I cannot share anything.
Hello.
Yeah.
Can you hear me?
Yeah, we can hear you back.
Okay.
Good.
Yeah. Thank you for waiting, by the way. It's sometimes like, these kind of technical glitches just come at that particular moment.
Yeah, sure. That's we have to account that always in the meeting. So please go ahead. And
so I hope you are able to see my screen.
Yeah, we are able to see your screen.
Are you?
Yeah.
Okay, just wondering. You are able to see my screen now too, right?
Yes. Some of you also confirm. And those who are not able to see have to pin his presentation.
And not any other screen.
We have to pin you. Yes, people are seeing. So yeah, so I can have another screen that you are people are we can see you. So I have multiple screens, so I can tell you that your screen is visible. Please go ahead.
Okay, good. So it's guys, it's quite easy. Like, go to change layout and go to tiled, like click on the tiled version, and then like one of the screen will be my screen. So just click on that and pin on that. So then you will be able to see my screen. Okay, Abhishek, are you here?
Yeah. Yeah, I'm listening to you.
Okay. Should I start?
Yeah, sure. So we have already introduced you.
Okay. So, and good afternoon to all of you. And first of all, I would like to thank Dr. Abhishek and the IOB team for organizing this international webinar on COVID-19 pandemic. And it's really a pleasure to be here and work and present our work on this diversity and evolution of coronaviruses. So here, in like today, I will be talking about the whole genomic and pangenomic perspective. So most of my presentation or research is focused on comparing the nucleotides like within the genomes, and then in the later half, like I will be more focused on the pangenome perspective of the whole genus beta coronaviruses. So we did this work with some of our MSc students from IBAB, and as you know, the reason we are doing this webinar and not like a real conference on site is the disease itself, like which we are discussing. And if you see, this is small virus, which is just 30 kb, like 30,000 nucleotides, has engulfed the whole world in its, infections, and it's continuously going on to do so until the vaccine will come. So let's hope the vaccine will come very soon, and I trust like all of you are staying safe and using all the precautions which have been suggested by the government and WHO. So this image has been made by Tanvi Kale, which is a MSc 08 student at IBAB, and I think it clearly depicts the thing that how coronavirus has engulfed the whole world in its pathogenesis. Okay, so before I start into the research part, let me introduce you where we are. So IBAB, it's a Institute of Bioinformatics and Applied Biotechnology, and we are located in south of the Bangalore. So, and we can say like we are just touching the Tamil Nadu border, and this is our main building, and we are well known for our incredible master's course, which is a dual specialization in bioinformatics and applied biotechnology. So we have a 50% focus on bioinformatics and applied and biotechnology. So the students, after doing masters, they get placed in industry. Many of them go in academia, and and along with our master's course, we also have PGDB course and research program, like PhD program, which attract a lot of students who wants to work within several dimensions of bioinformatics and biotechnology with like 17 faculties, which we are here. Okay, so I started working at IBAB in August 2019, and
I can't see the slides.
You cannot see the slides?
No.
Have you pinned the slides, sir?
Huh?
Have you pinned the slide? So you have to pin my presentation.
Actually, I can see only your face radiating there. That's all.
Okay. Abhishek, is it the problem with everyone? Just can you confirm?
No, no. Actually, people have to pin themselves to the presenter's slide, and then there are two profiles. One is Gaurav Sharma, another one is Gaurav Sharma in brackets presentation. So you have to take that one.
Yeah. Okay. I hope. So I joined in August 2019, and I remember that time, at that time we used to go out. So, but now that time is not here. So again, we will come to that. But so we have been working on microbial genome and evolution, and the model organism on which I have been working on from last 7-8 years is myxobacteria. So as I'm a microbial computational biologist, so I study their genomes, like how they. So these bacteria, they are soil-dwelling, non-pathogenic bacteria, which have a genome size of 9 to 16 MB. So that is around 3 to 4 times as of E. coli or bacillus, and this is a very large genome size as for a bacteria. And we are trying to understand that how these bacteria gained this much amount of DNA, and how their physiological properties are evolving with with time and as compared to their ancestors. Another aspect which I started working on like last 2-3 years back is we are trying to understand the signal transduction between different cousins of Vibrio. So like there are Vibrio cholera, Vibrio parahemolyticus, Vibrio fischeri. So we are trying to understand that how the signal transduction is different within these distinct Vibrio cousins. And another dimension which I started exploring, which I was planning to start, when I joined IBA, was to understand the total microbial community. So we want to understand how microbial communities are interacting with each other, how they are interacting with the host. So with a special focus on medicinal plants. But again, because of COVID-19 situation, everything is kind of on the hold. But I hope things will be better. Okay, so I would also like to tell you that why we started working on this coronaviruses. So as you remember, obviously we all are aware about SARS-CoV from January starting or mid of January. But in March 24th around, the government of India also announced that, there will be lockdown situation. So all the students were sent home and then all the faculties were asked to start teaching them. And I was teaching them advanced bioinformatics course, which is a practical course for me. So I thought that, it will be a good motivation, at least for students too, to start, if they start looking at these SARS coronavirus genomes. So that's why we started. And, I started giving them small lectures and small analysis to do. And then simultaneously, I thought that, okay, let's start doing something and maybe try to pose some new hypothesis or some interesting work. So let's start with the introduction of SARS-CoV, and I'm sure most of these things you are well aware of, and as other presenters have already said, and maybe based on your own knowledge too. So SARS-CoV-2, if you remember the Baltimore classification, so in Baltimore classification, they belongs to group 4, and group 4 includes these positive single-strand RNA, negative single-strand RNA. And if you talk about NCBI or normal taxonomy, they belong to genus beta coronaviruses. They are spherical and enveloped. They have around 30 kb positive sense RNA. Their RNA is 5' gap, 3' polyadenylated. They have 12 protein coding regions. So 12 protein coding regions, and the first two protein coding regions encode 16 proteins. So ideally, they are made up of 26 proteins. Okay, or they synthesize 26 proteins. And earlier it was considered to be animal to human transmission, but now human to human transmission is well known, as the officials all are saying you to wear mask. So what mask is doing is, instead of getting 100% probability of infection, reducing it to around 15% or 20%. So that's why mask wearing is very important if you are maybe in the vicinity of any infected person. And as you know about their epidemiology, it started from Wuhan, China, and now it's a pandemic. And their clinical symptoms, sometimes there may be no symptoms, and sometimes there might be severe pneumonia or fever, cough, shortness of breath. So as you can see, December 2019 to now, more than 9 million active cases have been reported, and around 500,000 of people have died because of this. And it is, it's a very short span. In a very short span of time, it has become a huge pandemic. If we talk about their taxonomy, they belongs to Ribovaria and Nidovirales. So Ribovaria and so Nidovirales, they are called Nido because of their meaning in Latin nest, because they make 3' coterminal nested sets of subgenomic viral mRNA, and then they belongs to family Coronaviridae, and they all are called coronavirus because of their appearance as solar corona under electron microscope. So if you see these Nidovirales members, some of them are spherical, some of them are like this, some of them are broad shaped, and different type of shapes are there. And then if we talk about the genus in which they fall is beta coronavirus, and this beta coronavirus is divided into Sarbecovirus and 4 other subgenus, and then we have 3 species in Sarbecovirus. So, if the molecular clock analysis, a lot of research has been going on these coronaviruses from at least last 20 years. And people have said that, the common ancestor of all these coronaviruses were around 81 8100 BC, and all these different types of coronaviruses, they start emerging around 3000 BCs. And just to give you a quick fact that the human coronaviruses are mostly present in alpha and beta coronavirus categories, and we will talk about this more. Let's talk a little bit about history of coronaviruses. So before 2012, the first occurrence of coronavirus was Murine coronavirus, and it was isolated in 1949, and it was well used for a lot of hepatitis and syphilitis case studies. And there were other animal coronaviruses too, which were pathogens for pigs, cows, and cats. And there were several diseases caused by these coronaviruses, but they were not becoming like a pandemic or even epidemic to some extent. And there were like a human OC4322E9, they causes respiratory disease. Some of the MHVs, they causes central nervous system, hepatitis, GI tract disease, and something like that. And if we just see a common, a timeline scale of these viruses, so we can see like, in after 1949, in 1960s around, OC43 and 22E9 was identified. Then in 2002, we got this SARS-CoV epidemic, and it affected like the whole Southeast Asia and several other countries because of the traveling and connectivity. And simultaneously, we also discovered HKU1, NL63, and after 10 years, we got MERS-CoV, again one other epidemic, which was largely constrained within Saudi Arabia. And then in 2019, we got this one. So basically, these three are the major pathogenic human coronaviruses. I hope there is some disturbance. Okay. So if we talk about their organization, so if you see family Coronaviridae, their genome sizes ranges from 25,000 to 31,000 nucleotides, and specifically beta coronaviruses, the gap becomes way smaller, like 29,000 to 31,000. And the important thing is the order of like replicase protein, spike, E, M, and N protein. So the order is always same with other accessory proteins popping up in between in different cases. So if we see more deeper into this, then we have ORF1A, ORF1B, which because the synthesis of proteins from ORF1B is halted sometimes because of because of not happening of ribosomal frame shifting, and then we have spike protein and all these different proteins. So in SARS-CoV-2 we have 12 protein coding ORFs. However, in SARS-CoV we have 14 protein coders, and as you can see, because of ribosomal shifting, all other proteins are formed. So basically 1A and 1B in all synthesize like 16 of these proteins. Okay, so this is about introduction, but when we start going more into, like research, we want to know like from where we should isolate the data. So the well-known thing is, I think, NCBI, and I'm sure all of you are aware of NCBI. This is the largest depository of all kinds of genomes, genes, proteins, literature, expression data, taxonomy, all kinds of data you can find it here. And one specific page is NCBI virus, which is dedicated for viruses, and you can identify most of the data, although I'm sure the most amount of data, at least for coronavirus, is available at GISAID. And another one is next strain. So here also we have a lot of genomic epidemiological data of at least coronaviruses. And then we have GISAID. So this is a very vast compilation of all the genomes. And right now, like I think it's 2 days only, that there are around 54,000 SARS-CoV strains available in here, and a lot of samples has been deposited here from India too. I think there are 761 samples as of 5 days back. Okay, and as Dr. Raja was saying, even Genotypic has submitted like around 100 strains. Gujarat Biotechnology Research Center also has submitted, CCMB, ICGEB, and a lot of other institutes have been providing data to here. And thanks to all of them that, all researchers all over the world are able to do a lot of genome analysis. And there is another page, which is known as virus pathogen resources. I'm sure like you might be aware of it too. And the good thing about this page is like, they also have a workbench and analysis and visualization tools, which you can explore in here. Okay, so what we wanted to do at that time, when we started doing this analysis, was to identify or explore the diversity at each level. So we wanted to identify the diversity at the suborder level, that how all the viruses within, suborders coronavirinae are with each other, then how the diversity is within beta coronaviruses, how the diversity is within Sarbecock viruses, and how the diversity is within SARS-CoV-2. So we want to identify their strain diversity, and especially for SARS-CoV-2, we want to identify their mutational hotspots. We want to identify their evolutionary origin, that how this pathogen might have evolved. Is it and another aspect which we want to look into was to identify the pan-genome analysis. So pan-genome is the total genome or total protein coding regions in all the genomes, and we want to identify that how it is shaping this genus and their members, and how it might be able to tell us about some novelty within this genus. So, regarding methodology, I would like to say that, we have used the standard bioinformatics tools, like for alignment, we use muscle plus omega, phylogeny, we use XML, and visualization with ITOL, and you can get all these details. Okay, so let's start exploring these things one by one. And I would like to emphasize here that our data source was NCBI at that time. And, we extracted all our data except one, which I will specifically point out, on March 29, 2020. Okay, so within family Coronaviridae, what we did, we took like 56 reference genomes. And, as you can see, there were 24 alpha, 19 beta, 10 delta, and 3 gamma. And we did their alignment using clustered omega, and then stripped off all the sites which were showing any gaps or something. Okay, so we used only conserved whole genome sequence alignment and make the phylogeny. So if you can see, these all are alpha, and these all are beta, these all are gamma, and these all are delta, and Wuhan WH1 is here along with SARS-CoV-1 here. Okay, and one important thing which I want to tell, at least to those people who don't know a lot about how to read the phylogeny. So we consider these branch lengths as an indicator of their distance. So if the branch length is higher, then it means that they are very distinct from each other. So if you see, like the members of this clade, they are making one single clade, and they are quite distant from gamma coronaviruses, and the outgroup is way far away from all of it. Okay, so the next thing which we want to see was that how all the strains within, all the reference strain within beta coronavirus genus is showing the behavior.
Sir, you are on mute, sir.
Okay. Now can you hear me? Sorry. Hello?
Yes, sir.
Yes, we can hear you.
Okay. Okay.
Yes.
Okay. Sorry about that. So now we want to understand more about subgenus cercovirus. And so for this analysis, we took like 420 420 genomes only. Okay. And the distribution was like 103 was from SARS-CoV-2, 312 was from SARS-CoV, and 5 from pangolin. So my mistake. This is not reference. These all are normal submitted genomes along with one reference. Okay, and then we did the alignment and used the conserved whole genome sequence alignment and generated the phylogenetic. And again, I'm extremely sorry for these bulky images, but I will try to explain them to you in a way that it helps you. So if you see, so all these blue indicators, they are SARS-CoV-2 strains, and the red ones here and here, they all are SARS-CoV, and the green one, which you might be able to see here, are pangolin. So if you see here, all these SARS-CoV-2, they are making one single monophyletic clade of themselves, and the next closest homologue is RATG13. Okay, and as Dr. Urmila also told like day before yesterday, and we also got the same results here, that RATG13 is the closest neighbor, and then followed by pangolin covey. And the next two were again from bat. And why I'm pointing it out is because they, like at least again, as you remember, I told that, branch length determine how close or far they will be. So if you see, this is like all of these are making one single clade without any with very few differences. And RATG1 is a little bit distant from all these SARS-CoV-2 strains, but pangolin is a little bit more far away as compared to RATG strains. And similarly, the next two strains, China COVID C45 and COVID XC21, they are also a little bit far. So basically what we can say is that the SARS-CoV and RATG and pangolin strain, they all share a common ancestor, and they might have got diverged way earlier. Maybe we don't know the time frame right now, but they might have got diverged years ago, and now we are really seeing the infection caused by SARS-CoV-2 in humans because of just a random chance. Okay. Another thing which we want to analyze was now to see the diversity within SARS-CoV-2 strains. So for this analysis, we used 167 genomes and again used the same strategy. And as I told you that, we extracted the information from NCBI as on March 29. So it is quite a way ago. And at that time, there were only 2 Indian strains in there at that time on NCBI. I'm sure on GISAID, the situation would have been different, but I was not looking at that right now, at that time. And so we used the alignment and generated the phylogeny. And what we identified was that, that there are several country-specific clades, as you can see here. So if you see, all these strains are from USA, Washington, this whole clade, or maybe the things will be more clear in the next slide. So all these strains belongs to USA, Washington, and similarly we have another USA clade here, okay, belonging to the same isolation like Washington state. So, it clearly indicates that there are more than one specific clades of, these strains. Even for India, we found one strain here and one strain here. And because our data set is quite, I will say old at present time. So we propose that, that there might be different types of strain, moving around and causing infection across, even in the same city, state, or country. Another thing which we also noticed was that the China Wuhan strain is very similar to kind the USA Cruz strains, which which is the strain isolated from the Cruz, on which there were a lot of infections, and which suggests that that they might have got it from the Wuhan seafood market. Okay, so another aspect which we were most interested was identifying mutational hotspot or at least mutation sites. So we used the same data set 167 reference strains, and we did the analysis. And what we found was that there were several sites. So let's talk about what we did. So we took the reference was Wuhan HU1 strain. We aligned all our genomes with Wuhan HU1, and then for each site we try to identify what is the percentage distribution of of any particular nucleotide. Okay, so let's say on site 8782 we have cytosine. So on 8782, the chances of getting cytosine was 65% and getting thymine was 34%. So we noted that and tried to make a heat plot like this. Okay, it's not really a heat plot, but just to document it. And we did it for all the locations. And then we identified that which sites have a very high mutational probability rate as compared to others. So overall, we identified that there are 290 sites which have a mutational probability. So besides 290 sites, there were only 290 sites which were showing a mutation or either AGCT to AGCT, any combination. Okay. And we haven't, because I'm sure a lot of you know about genomics. So when you submit a genome, sometimes there are gaps in between and they try to fill it with N or some other random non-traditional nucleotides. So we haven't considered those sites as a mutation. So we haven't covered them in here. So then what we tried to do was to distribute them in 4 different categories. So high mutational probability site, which was showing more than 19%, and these are just random numbers. Okay, so there were nothing to follow the standard. So we identified that that more than 19%, there were 5 sites which were showing mutation at their particular site, and similarly the medium 4 to 19% probability, we found 10 sites as can be seen here, and then we found 28 sites with range from 2 to 4%, and we are talking about 167 reference genomes. Okay, so if we are saying that let's say here we are saying 35% mutational probability, it means around 55 genomes have thymine, and rest of the 2/3 have cytosine. Okay, so that's a lot of genomes or lot of strains which we are talking about. Okay, so and if you see, many of these sites are a part of coding region. So 8782 is part of ORF1A, and it is changing from C to T. And ORF8, similarly, we found one site on spike protein, which is having 11% mutation probability, which is from A to G. And another thing which we did was to understand either it's a transition mutation or transversion mutation. Okay, so we found that out of total 290 around, there were 158 sites which were transition sites, and there were 86 sites which were transversal sites. So I'm sure you guys have read about it, that transitions are more of a silent mutation. They lead to a silent mutation. Whereas transversions, they make real mutations. So they change the protein structure, they might change the protein sequence, structure, and their function too. So we suggested that people should be more focused on these transversion events as compared to the transition events. And the probability of having a transition event and as compared to transition event is almost half as compared to each other. Okay, another thing, so again, remember this all data is from March 29th, but for this work, we want to understand that how mutation might be evolving, or how any mutation site might be evolving. And I would like to say here that we used so now the data source is GIS AID, and we extracted this information on June 19th, like just I think 7 days ago, and we got like around 717 strains at that time, which all only from India. So right now we are only talking about Indian strains. And if you see the distribution, so what we did, we distributed these strains into 4 categories. So based on their sample collection. So so till March, there were 137 strains. In April, before strains were submitted, in May, 268 samples were collected. In June, till now, at least till then, only 51 sequences were submitted. Okay, and then we want to understand that in each month, or in each of these month sample, how the mutations are changing, or which are the high probable mutation sites. Okay, so let's see. So we did the same analysis as I have shown here. Okay, and then this time we try to generate a heat map. So the heat map is showing, the colors are here, like the maximum mutational probability we got was 0.49. Okay, so it means that 50% chance were that at that particular site there were a different nucleotide as compared to the first one. Okay, so and we generated this heat map, and you can see we found like 5 clusters: 1, 2, 3, 4, 5. Okay, and this is in March, this is in April, this is in May, and this is in June. Okay, so I hope you guys can clearly see this, that at least in this cluster, the mutational probability of these sites were higher in April, but later on in June, at least according to our this independent data. So I will say, because on GIS AID, the data is from all places, from all institutes, or there is no dependency here. So we are just talking about this independent set of data, that how they might be behaving. Okay, so the mutations were higher in, like these mutation sites were having mutation in April, but in June, the numbers have gone like really sharply down. And if you see in the contrast in here, the numbers were, these sites were not mutating as such in March or April, but in May and June, the mutation probability is exponentially increasing. So what we did, we generated these trend charts. Okay, so as you can see, the maximum is 0.49 around here, or maybe here, I'm not sure. But if you see in the first cluster, where we have 9 sites, and I will discuss those sites, there was a sharp increase in April, and then later on, those probabilities got decreased. In second one, it was kind of almost like staying same throughout all these 4 months. In these 3, it was kind of exponentially or linear kind of increase. Okay, so again, we try to see their expression in in the mutation probability, like 1 by 1. And again, as I told you, that transition events may be silent mutations, but transversion events might be like the real thing. Okay, so we found like one of these G2T, which is an ORF3A, and earlier it was only showing 0.05, then it started 0.17, then 0.28, and now 0.37. So it is, and these 17 sites were the major determining ones, at least time period wise. Okay. And we will try to, this is just a recent data, so we will try to, compare them with the total average of all samples, and then see, are they like really behaving, like, are they the really trendsetters of mutation in these viruses, or how they are behaving. We will start looking more into this kind of analysis. And I almost forget that these figures, they were made by Ajra, and one of my master's second year student, and they have just finished their cases. So the next thing which we want to know, and I hope, Abhishek, I'm not running out of time, right?
Can probably you can, if you can finish in 3-4 minutes, that will be great.
Okay, I will try that. Okay, so we want to understand that how what is the origin of this SARS-CoV-2? Okay, so what we did, we used a different strategy as compared to others, or I'm not sure, like we used this strategy that we segmented, we took 4 reference genomes, SARS-CoV-2, COVID, pangolin, and MERS. MERS was just as a standard one because we already know about it, like everything, and SARS-CoV-2. And so we segmented each genome into 100 nucleotide, and then did the BLAST and against NCBI virus database, again April 6 downloaded, and then we removed the self hits first. So if we are talking about SARS-CoV-2, we removed all the hits which were from SARS-CoV-2, and then extracted top 50 hits and identified the host representation and made a heat map. And we also took the best hit and represented it, that, and this is the closest hit. And I'm extremely sorry again for a bulky image here, which might be a little bit tricky to understand, but I will again try my best to convey like what we want to say. So this is MERS-CoV, this is SARS-CoV, this is pangolin, and this is SARS-CoV-2. Okay, so let's talk about MERS-CoV, and these are 1 to 100, 100, 200, 200, 300, like this throughout the genome. Okay, so if you see the closest one, so the closest one was because we removed the self-hit MERS-CoV human samples. So we got the first best hit in camel, and as you can see, all of them are showing very high similarity with camel. So so camel was the intermediate host for MERS-CoV infection. But if we remove the camel from our data set, then what we are getting is that bat coronaviruses are having the maximum homology throughout the genome, and there were several unique reasons which we were not able to find in any other organism. Okay, so that is for MERS. Now, if we see about SARS-CoV, so SARS-CoV again, the closest is bat, and then so if you see in SARS-CoV, we have most representation, like the first hit was in bat, but in between there were some reasons which were very specific to SARS-CoV, which were not showing any homology in any other host. And similarly, we found that throughout it was bad, but in between there were few sites which were very specific to SARS-CoV. And if you remove, if total role distribution, bat is the maximum one, but a few of them were showing, obviously because some of the parts are similar to COVID-2, so they also have some homologs in COVID. Okay, and now if we see about SARS-CoV-2, we found that. So if you see this overall picture in the last circle, most of the representation is in bat, as you can see. But in between, if you see the green region here, so this green is denoting pangolin. Okay, and there are several sites throughout the genome which is showing more homology with pangolin as compared to bat. And there were some other sites like this one, which were more similar to human COVID strains. Okay, and some of them were unique too. Okay, so basically what we are concluding from our analysis that the SARS-CoV genome has a vast amount of similarity with the bat coronaviruses, but in between there are several sites which are very specific for pangolin, and many of these sites are very unique as compared to other hosts. Okay. And the next thing, and the final thing which we want to discuss was the pangenome. So, if you are not familiar with the pangenome concept, the pangenome means that in a closely related organisms, what is the total protein coding regions which are similar? What is the total protein coding? Sorry, what is the common protein code? Not the common is not the problem. What is the overall depiction of total protein content? Okay, so it is made up of core genome, core proteome, which means those proteins will be conserved across all of them. There will be unique proteome, which will be present only in one species, and then there will be accessory proteome, which will be present in multiple combinations of species. So what we did, we took 19 reference genomes of beta coronaviruses and I extracted their protein coding regions, and then did the ortholog identification using reciprocal blast, and then identified the pangenome. So if you remember, like in all these genus beta coronaviruses, the proteins are ranging from like 5 to 14. So 14 is the max in SARS-CoV-1, and 12 is another type of these beta coronaviruses, RNA-seq. Okay, so we identified that MNS protein, they are always well conserved across all of them. ORF1AB is conserved across all, but in MHVA59 strain, it's not there. And another thing which I would like to point out here is that all these annotations have been taken from NCBI directly. So, and if you see E protein, envelope protein, it's present in all of them, but in here, we didn't find a respective homologue, but a different kind of envelope protein were present in here, like small envelope protein, which were present in these novacoviruses. And so this is the core proteome, which is conserved across all of them, and this is the accessory proteome, and this is the unique proteome. So if you see, ORF10 is unique to SARS-CoV-2, and these 4 proteins, SARS-3b, SARS-8a, SARS-8b, and 9b, they are conserved. They are only present in SARS-1. In pangolin, we didn't find any of these any unique protein. Okay. And so this work has been, this figure has been generated by Kishan, another one of my MSc student, and we also tried to portray it using a different strategy, and Neeraj, another student, made this figure. So if you see, and it also helped us to give like different hypotheses too, like these proteins, they are only present in Mbecovirus. These proteins, they are only present in these Marbecovirus. And similarly, there were different combinations like these ones, they are only present in SARS-1, and similarly, something like that. So with this, and another thing which we want to know, that how this pangenome is behaving across all these genomes. So we identified, so when we plot this pangenome for different combinations, we found that it is continuously increasing with the addition of a new species, which means that it's an open kind of pangenome, which suggests that the pangenome with the input of any new species in this cluster, the pangenome will keep increasing, and it's not closed, which means there are, or there might be more novel kind of viruses which might emerge in this group and can lead to infection and behave differently. And all this is happening because of the unique proteome in each of these genomes. So if you see, this is the pangenome trend, and this is the unique proteome trend, and they are continuously increasing. Okay, so with this, I would like to just finish it off then with some take-home message. And so one of the main messages that SARS-CoV-2 strains are very different from SARS-CoV-1 strains, and they are clearly making a single monophyletic clade as compared to others. And they are closest to RATG13, followed by pangolin, reasserting the fact that they might have evolved from a common ancestor, which was putatively resulting in patopangolin. And we also identified that there are several clades for each country, which means that there are multiple strains causing infections in each country. We identified like 43 mutation sites with high, medium, and low probability, and they might be the determining factor of the making of new type of strains. And another thing was that, as we know that virus genomes are constantly mutating, we identified several sites where mutational probability is emerging with each month, whereas at other sites they were relapsing back to the normal. So they are constantly like the mutations at one particular site might be going up and down depending on their uses, that are they really helpful for the virus or not. And we identified the pangenome and suggested that the open nature of this pangenome implies that the genus beta coronavirus has an unlimited large gene repertoire, and with the addition of new type of viruses, their pangenome will keep increasing. And there is a lot of novel kind of viruses which are yet to come. Okay, so with this, I would like to thank DST and Department of Science and Technology and DST Inspire Faculty Fellowship Award, and along with the IBAB for supporting me and providing me with the infrastructure. And this is a wonderful group of people who are working with me. And these are MSc first year students. They all are in the lockdown right now, and away from here, but they really helped a lot in making this work. And Ajra, she also did some work in mutation. And with that, I would like to thank you all for being a patient listener. And yeah, I would be very happy to take any. Hey, Abhishek, can you hear me?
Yeah. So thank you, Gaurav, for a nice talk. And if there are some questions, we'll take on. Let's see if there's a question. So
if there is a question, like how many of the high probability mutations are in protein coding site? So as I have shown here, that there were several mutations which were in, like, even if we see about this how mutations are evolving status, there were most of these mutations were in the coding sites only. Okay, like the first one is in the non-coding site. I hope you are able to see my presentation, right?
Yeah.
Yeah. So Sandhya, the question of Sandhya is that are they in coding site or non-coding site? So some of them are in non-coding sites too, but most of them are in the coding site.
Okay, so please keep your questions.
Okay. A Singh is saying that can it be generated using software like Mega? Yeah. Phylogeny, there are different phylogenetic tools which you can use. Mega is one of them. It's easy to use because it's Windows based and obviously along with Linux based too. So Mega can be used too.
Yeah. So basically, one of the speaker has some urgency. So
Okay.
What we will do that you can keep asking Gaurav question by email, and Professor Dr. Yusuf Malik has been waiting for his talk. So he just requested that can we make his talk earlier. Then
Okay.
I will keep my talk for later, and also Dr. Tikam's talk because the guest is from Israel and he probably have another meeting. So let us invite Professor Yusuf Malik for as a next speaker. Thank you, Gaurav, first of all, and we'll be in touch. Please welcome Professor Yusuf Malik now for his talk. And Professor Yusuf Malik is a professor in Israel, and he's working in machine learning based bioinformatic methods. And he will discuss about his work on machine learning methods for SARS-CoV-2. So please, Professor Yusuf Malik, please go ahead.
Okay. Thank you for introducing me. Also, I would like to introduce Shlomo Izhar Ben Shlomo.
Yeah.
Is also present here. We're gonna present work. I am a computational guy and he's the medical guy. So we're doing a collaboration in different projects. And actually, I would like to start maybe by Izhar telling us the story, how we got to this project. Then I proceed, and then we continue this in this way.
Yes,
so Izhar, can you tell the story?
The story goes like this. I was talking to my neighbor, who is not a doctor, nothing. He knows nothing of medicine. And he simply dropped me the question is, do all these vaccines that we get as children, don't they help us against this new disease? And I looked at him and I said, you know what? Honestly, I cannot give you an answer on this. But I have a good friend. His name is Malek Youssef. He knows how to mingle with this. And then we started discussing it. And what is presented is actually the outcome of this common effort after the seemingly stupid question of my neighbor. That's all. Please, Malik. Please.
Okay, so in order to answer this question, actually, so the question was, as Izhar described it, is all this vaccine that we took all over our life is somehow could be helpful to fight corona or COVID-19. Then what we have done, actually, we went to this public data, the WHO coronavirus disease dashboard, which actually report about daily about the COVID in each country. And also we luckily find all this information in the same website, but in another location that report about the vaccine coverage over years for every country. If I would like to give you some info, we go to this, for example, this is actually the website that actually you can access it to get information about the vaccine coverage. And if you just pick here and let's see, press here, then you see the coverage for this specific vaccine over a couple of years. Actually, it started by 2008. And this is actually the percentage of cover, 97%, 91%. This is for each country for this specific for this specific vaccine. So the question is how we can integrate these two data sets together in order to answer the question that raised by Haeußler neighbor. Actually, so I will explain one by one how we get to the data ready to be used for machine learning, and then Haeußler will continue to explain the outcome. Because, as I said, I'm a computational guy, that I just understand how to work with data. So actually, this data, as I just show you to you, is look like that. So in order to bring it together, we were supposed to write some script to read all this Excel file, or actually it was a big Excel file that described for each vaccine over years, over countries, and just we need to summarize it somehow like that. Then actually we have this nice table that explain in general. Here we have the country name, here we have the name of the vaccine, and this is the average of the vaccine, the coverage vaccine. I think it's the last 10 years, yes. So this is the numbers that we have got from this data analysis. And okay, just keep it in your mind that we have this nice table. Then the question, how we compare this data with the COVID data. So then we went to the coronavirus disease, COVID-19 dashboard, and we download the recent data. Actually, we download the 3 times the recent data. And then actually also we process the data in order to come up with this nice table. This is summarized table that give for each country the cumulative death, cumulative confirmed. And then we said, okay, so let assign if the ratio between the cumulative death and cumulative confirmed is less than 0.3 or 2 or 1, we try different threshold, then we consider this country as positive. That means this country somehow, the effect of the coronavirus, the effect of the coronavirus is not so strong, so it hit it less than other countries. So we decide to give an class label as positive, and the other we give it a sign negative. So in this way, actually, we produce a data with label. Okay. Then the question, how we can combine the 2 data sets. Actually, we combine it. Now we have the whole data set, the data from the vaccine that we summarize it in this way, the data from the current situation of the death and the infected and confirmed cumulative confirmed. So then we have this data ready for machine learning. If you guys familiar with machine learning, in order to be able to run the machine learning supervised algorithm, you're supposed to have a table or data that you have the label of the example and you have the features. So the features here or the attributes actually is the vaccine, And the label actually is the ratio and the ratio that we calculate here. So positive, that means the country is somehow in good position. Negative, the country relatively is in bad situation. So assume that we would like to find if there is a correlation or connection between these vaccines values and the label. So this is actually a traditional machine learning approach to find the correlation or to predict actually the label of the country according to these values. And in order to do that, we use K9, which is a very popular machine learning or data science tools that let you also make your own programming. So we read the data, we partition the data into training testing. The training part goes to the learner. Actually, we use decision tree and random forest, and we hide the training part, the testing part from the learner in order to test our model. So in this way, we can see if actually we have create a model that can able to predict the label of the country. So we repeat that 100 times, and we actually in the end we make a summary and aggregate all the results and present it. And actually we can also see the model as a decision tree. So this is actually the workflow that we use it. Actually, the one that we use it is more complicated, but this is the simple one in order to explain the idea about our approach. Okay, so then actually we try different MR threshold. As I said, the MR actually is this ratio. We try 1%, 2%, 3%, and this is actually the number of positive, the countries that actually apply this threshold or satisfy this threshold. Negative 43. I would like to say that we also remove all the countries that report less than 1,000 cases. In order, we think there is some, some effect of this maybe fake report. So in order to do that, we just remove all these noisy countries from our analysis. And this is the performance, the accuracy, the record, sensitivity, specificity, precision, and F1 measure. Actually, if we look to the accuracy, for 1%, we have very good result association. This is the average of 100 iteration. That means that result says that there is a connection between these numbers, the vaccine coverage, and the label of the country. The label of the country is actually the ratio between the cumulative death and the cumulative confirmed. And if you look to the 2%, then the accuracy somehow also look nice. 3%, then we also getting this result, but sensitivity goes worse. And in order to validate more our results, we just shuffle the label of the data. That means we just shuffle this column in order to see if the result is still valid. And we run it again, and you see the performance is very, very low. That means the label is somehow connected to these numbers. And after that, in April 24th, we again download the data from the dashboard of COVID-19 reported by the countries and published in the WHO website. And we see, again, we see the same behavior again after actually 13 days. And again, also we try in the June 3, actually this one is somehow cut it. And again, we see somehow good results. This is actually the top features. As I said, the features actually is the vaccine. And this is the importance for each vaccine in our model. So I think this is I think this is the time that is how to explain the meaning of this table and the conclusion.
The important thing that actually pops up to the eye is the fact that all the 5 first vaccines are vaccines that are typically given to children and are almost completed during the first year of life. And a study which we don't cite here because it was published in Hebrew on the lay press and on the internet, is that in a very crowded town where there were many, many cases, they are living in very crowded conditions, and they made an in-depth analysis, and they found that children up to the age of 10 are less readily infected, less readily infect others, and are ill much on a much lower level than adults. But interestingly enough, up to the age of 1 year, they are contracting the infection just like adults. So in some way, it speaks to the validation of this observation. Now, it still remains a big question how vaccinations that are mainly given during the last 40, 50 years affect mortality rates in generally older people. But still, the connection exists, and it's for us and others to try to explain it. This is amazing. And also look at this, that hepatitis B, third immunization event is important at a level of 100%. The next one is only 0.377. So this is amazing. And this hepatitis B vaccination and rubella containing vaccine, Malek, you have to enlarge it.
Yes.
Anyhow, this is amazing. And Malek, can you go to the tree of decisions?
Okay.
If we translate it to tree of decisions, then you see that hepatitis B third event of vaccination, and actually the level of compliance with the vaccine, the coverage of the vaccine, puts 34 out of the 35 negative countries to the left side. It's amazing. It's so striking. And then the rest, the 27 countries that go, if you divide them by polio-containing vaccine, then again, they divide into two distinct clades. The left one says that by HPV to females, this is something that sneaked in. Again, you can get the final determination, the final leaves of the tree. So this is striking, and there's a lot to investigate about this connection. And the last slide would leave us with the questions to the future and things that people should be doing in the future. Malik, can you give me the last slide, please? Okay, what can we do next? Our first thought, we don't have access to these databases, but we should review personal vaccination schedules of people who were infected and people who were not infected. Maybe those more readily infected lack some vaccinations on younger age. And moreover, if children, very rarely, but children and young people got the disease on a higher level and more severe disease, check the severity of the disease against their personal record of vaccination. You might discover that indeed this may matter, and this might be very, very important. And the last thing is, we were thinking about taking a few kids who are 9, 10 years old, take their sera and their immune cells from the blood, and verify in the lab whether the sera of young children, which are naive to the coronavirus, they were not infected, they don't have antibodies, specific antibodies against the virus, but and also some people who are older than 50 years, and see whether the naive sera of these two groups react differently to proteins from the virus. Because if this is true, then basically we are looking at something hate to think about. And this is the possibility that there is a cross reactivity between viruses that do not necessarily belong to the same family or the same clade, and they are really distant. But again, if you look at a child that was vaccinated intensively during his first year, with all the schedule that is now accepted as standard in many countries, it might be that this is a major source of protection. And the thing that might come up from this is, indeed, if this is the case, why not go and vaccinate people who are 50 or 60 years old, not even against this COVID-19 guy, but against probably the next guy that might pop up in the next decade. We are looking at an unknown future with viruses that spread like hell in our very communicative world. And we might be looking at something that people didn't think about. That's it, but there are more questions than answers. Thank you.
Okay, actually, thanks, Izhar. I think we'll be happy also to discuss more ideas, get your feedback, because actually it's not easy to convince people that this approach is valid. So we'll be happy.
Yeah, thank you, thank you, Malik ji and Doctor, for coming up together and presenting. It was a really nice presentation, and floor is open for some questions. And we really thank your team, Professor Yusuf Malik and Professor Izhar Ben Shlomo. Yeah, we are.
And we'll be also happy to collaborate. If anybody of the people who are present here will think about collaborating with us, we'll be open to that. This is amazing. We like it.
So one of our speaker, Pras, has the question. Pras, you can raise the question by opening your mic. So.
Oh my God, this is fun. I think the predictions for longitudinal study would make more sense. For example, over the period of 10 years now, how many patients have taken the drug per se and then checking among them. The question is to take a longitudinal study. Always a longitudinal study is good. But with the COVID-19, we are facing something that is a cross-sectional, because time is still ahead. But basically, the people are taking to vaccines, to those who are not physicians. Vaccines are taken in up to 3 or 4 times during childhood, according to the table, to the schedule. They are not taken later. They're just taken during the first and second years of life, and that's it. So I would not be able to tell if somebody continues to use the vaccine later. Nobody is actually being vaccinated just for the fun of it. So, okay, that's my question, my answer to the question about longitudinal study. Go backward with the longitudinal study. The most promising thing is to look into the individual vaccination history of people who were infected against people who were not infected in the same community with probably the same, and look and see if this guy really missed one of the vaccinations in childhood, he might be more prone to develop the disease. This is the way we think, but again, we'll be glad to team up with someone who has this sort of database and work together on the things. Thank you in India.
Thank you, sir.
It's fun to be in touch with people.
Yes, sure. We are going to organize another meeting separately on Indo-Israeli cooperation, and that will bring us more thought-provoking discussion of various kinds, and we are indeed planning for that actually. So
okay, great. So we'll be delighted to collaborate with whoever wishes to go on the lines and suggest a new line. Okay?
Yeah, thank you, sir.
Good. Have a good morning to you and good afternoon for us.
It's a good evening
Bye-bye.
for us and good afternoon for you. So,
Thank you very much. Goodbye.
bye. Thank you,
Bye-bye.
sir. Thank you. So with this, we have come up with one more presentation, and since I am presenting as a local, if our next presenter, Dr. Jan is available, probably he can present before me. But since he's in USA and we had promised him to last talk, so he has to come around 6 o'clock, 6 or 7. So probably he's not available. So then I will present. But if he's there, then I will ask him to present. Miten Jain, are you there? Dr. Miten Jain?
Dr. Abhishek,
Yeah.
this is Dr. Tikam.
Oh yeah, Tikam's turn is still there. Oh, sorry. So Dr. Tikam is assistant professor in university in Udaipur, and he has several years of postdocs in USA and Canada, if I'm right, and he is also working on coronaviruses. And somebody has okay, you are expert guy. So he's as you said, Mohanlal Sukhadia University, Udaipur, Rajasthan. Udaipur is a beautiful city, and he's working on coronaviruses, and I invite him for the talk. And please go ahead, sir.
Thank you, Dr. Abhishek. Yeah, hello.
Yeah, please.
Yeah, okay, just a minute. Yeah. So, are you able to hear me, everyone? Hello? Dr. Abhishek, are you able to hear me?
Yes, I am able to hear you.
Okay, so thank you, Dr. Abhishek and Institute of Bioinformatics for giving me the opportunity to present the work. Basically, I have been working on the coronavirus since 5-6 months, as you all also working on the coronavirus. So today I am giving a presentation on synthetic RGD peptide-based therapy for coronavirus treatment, COVID-19 treatment. And the content of my presentation is the introduction of the SARS-CoV-2, drug repurposing, and the EDT chelation therapy and RGD peptide, synthetic peptide-based therapy. So Dr. Abhishek has offered me only 15 minutes for this presentation. So I have also seen that most of the things about the genomics and the drug discovery of drug discovery related.
Your screen is lacking the PPT. It's only P and int we are able to see. So you have probably have to turn it to presentation. So it's partly coming actually.
No, it's coming or not? Yeah.
Now it's looking. Yeah. Now it's
okay. So most of the things has already been covered by the other speakers, and about the genomics part of the coronavirus and the structural biology part of the coronavirus, and the drug discovery related part about the coronavirus. So many of we are also very delighted to know that many of the new sequences has also been submitted by many Indian scientists, and I really congratulate them, and I really thank them for such very nice, really nice work. So basically, the coronavirus is really a very dangerous virus. So the students or the kids, they cannot go outside to play. We cannot go out in the streets. People, they cannot marry, and everything we have to do online mode, school fees, rent, payment, everything we have to do online. And my story is something different, that, okay, I'm taking the online classes and the students, they are just sleeping. And so, you can see that this situation is becoming like hell. Sorry for my word, but it's really senseful word. So, but anyways, we have to adhere to some government guidelines, and we have to practice social distancing, and we have to stay home to be safe. So this is basically the brief of the coronavirus. So if I start with the microbiology part, I would not going into the details of the microbiology, but just because I have already seen that most of these presenters, they have already mentioned many things about the microbiology and other things also. So I would be just skipping the part which have already been dealt, and I would be only give a brief idea from my side only. So it's the coronavirus that is basically belong to the nidovirus, and we have alpha coronavirus, we have beta coronavirus, we have gamma coronavirus, and we have delta coronavirus. And our SARS-CoV-2 belongs to the coronavirus, beta coronavirus. So if I talk about the natural history of the coronavirus, it's this virus, coronavirus, it's not the case that we are, for the first time, we are getting infected by this kind of virus. It's probably starting from the 1930s, when we had the infectious IBV virus. So, and then in 1940, 65, and 2003, 2004, we had the SARS coronavirus 1. So, the thing is that, so this coronavirus is not a new virus, but of course, yes, the SARS coronavirus 2, it's coming up with a very different genotype. It's coming with a very different genetics, and it has the infectivity and the mortality very, very high as compared to the other known coronavirus. That's why some people, they have termed this virus as the novel coronavirus, but this is not something new. We had already experienced a lot of coronavirus in the past. So the outbreak took place in China, and subsequently it spread into the other parts of the world also. And in India, we had the infection, the first case of infection in January 2020. So this is the situation. Currently, we have the situation all over the world. This is basically called as a pandemic. So it's the whole world is now getting affected by the coronavirus. Oh God, please mercy on us, really. So if we talk about the numbers, so if I talk about the global numbers, so we have more than 7 million cases of infections right now, and we have more than 4 lakh people already died of this disease. So it's a very, very difficult situation. And if you make a comparison, as I already mentioned a few minutes ago also, that the SARS coronavirus whose outbreak took place in 2003, 2004. The SARS coronavirus infections last for 8 months, and it affected almost like 26 countries, and 8,000 people got infected, and 700 people died approximately. But if we talk about the SARS coronavirus 2, within the 6 months, we have more than 200 countries affected, and 7 million people already, more than 7 million people infected, and more than 4 lakh people already died, and we are still counting. So the situation is, we can clearly see that this virus is really, the infectivity of this virus is really very, very high. And but more or less, so we have published a lot of work also, but until now also we are not able to understand what are the main causes of its infectivity. So basically, I'm also coming up with some new thoughts and some new insight into the higher infectivity of this coronavirus. So basically, the coronavirus genome has already been discussed by many other speakers also, and I am only interested in the one of the 4 structural protein that is basically the spike protein. So the spike protein is the protein that is the first protein that interacts with the host cell receptors. So if we say that, okay, what's in the virus that is first making contact with the human cells, that basically the spike protein, and that's why it becomes very important to study the the spike protein first. So it is the spike protein that make the interaction with the ACE2 receptor and then the nuclear material of the virus that goes inside, and the virus it hijacks the human system, and it produces the new virus for the multiple round of infections. So that is basically the multiplication of virus and everything. If we see about the immune response of the SARS coronavirus is the same as we have the other viruses also. But yes, of course, the severity is very high, the infectivity is very high, and the viruses, SARS coronavirus is using a multifaceted mechanism to elicit the immune response in many different ways. So because of this, we have an unleashed cytokine secretion in case of COVID-19, and these cytokines, they go to the different parts of the body. They affect the multiple organs, then we have the multiple organ failures. And this situation becomes worse in the immunocompromised people and the aged people. So if we talk about the symptoms, so I do not want to go more into the details about the symptoms, but we all know that the symptoms are fever, cough, and malaria. We know that nowadays the situation is that you may be COVID positive, but you may not be showing any sign of COVID, getting coronavirus infected. So you may be asymptomatic or you may be symptomatic also. So that's basically a real challenge nowadays, that many people are symptomatic and many people are asymptomatic, and both are COVID positive. So the situation now is that this is just the conclusion of my first part, it's not the conclusion of the whole presentation, that the coronavirus has high infectivity, although low mortality, 6 to 7% globally, and if you consider the Indian data, it's only 2 to 3% of the mortality. So the disease display a complex etiology and pathophysiological outcomes, clinical signs and symptoms. What I was talking about, that many of the infected people, they may have the symptoms, many do not have. Many have different types of symptoms. So it becomes difficult for us also to understand whether we are really infected or not, and also we can choose the doctors also whether we are infected or not. So the thing is that the take-home message from the first part of my presentation, that the SARS coronavirus is very, really, very dangerous. So if we talk about let's proceed for the second part. It's a drug repurposing part. So many people, like when I started doing the research, many people asked me that, okay, it's not the case that we are getting infected by the SARS coronavirus for the first time. So what is the main reason why the previous medicines or whatever the antibodies or neutralizing antibodies that were raised against the SARS coronavirus, they are not effective for the SARS coronavirus too? That's basically the reason that we are not able to use the old medicines for the treatment of SARS coronavirus too. And for this reason, what we are doing, we are just using the known tactics to somehow curb this disease or to combat this disease. We are using the chloroquine, we are using hydroxychloroquine, we are using a combination of some antiviral drug and chloroquine. Like that, we are doing many things. We are also using the plasma therapy, but the thing is that we do not have any drug right now, we do not have any medicine, we do not have any vaccine. So, and even if you see here, there are many drugs and there are many vaccines, many things that are in pipeline, but it would take a lot of time. And if you talk about the drug discovery, you can see there are many components of the drug discovery and development: target identification, validation, hit identification, hit to lead optimization, then candidate identification, and then if you go for the development, large scale development, commercialization of the product, licensing, it takes almost 15 years. So if we do drug repurposing, in that case, we can save some time. We can finish everything in 2 to 5 years, 3 years, 4 years, 5 years. But of course, yes, that in drug discovery we have some opportunities. So this was a very small part, and I intentionally make this part very small because that was not much related with my presentation. It has this very little relevance. So the take-home message from this part is that in the mid of the pandemic situation, we have nothing available in our hand. No drug, no vaccine, nothing. And for everything we need time, and the coronavirus is really not giving us time. It is continuously killing the people. It is continuously infecting the people. So what I have done in the research is the part third, which I would be more interested in talking in detail, is the pulmonary EGT chelation therapy and the synthetic RGD peptide-based therapy for the coronavirus treatment. So as we all know that I have already also shown in my previous slide that it's basically the spike protein that come in contact with the receptor protein of the human cells, and this virus get its entry into the host cells. This is the case of the lung epithelial cells. So what I did, because I already mentioned that it's the spike protein of the coronavirus that is coming in contact with the human cells. It's the first protein of the virus. So what I did, I compared the spike protein of the SARS coronavirus 2 and the SARS coronavirus 1, and I tried to see what are the differences, where are the differences. So what I found in the SARS coronavirus 2 is that the SARS coronavirus 2 has an important functional motif that is called as RGD motif. And that RGD motif is not present in SARS coronavirus 1, and it is also not present in any other SARS coronavirus. So the importance of this RGD motif is that this RGD motif was originally found in fibronectin, and fibronectin is an extracellular matrix protein that has a role in cell adhesion and the cell attachment. And this RGD motif is also found in vitronectin, and it is also present in laminin. So the commercial application of this RGD motif is that we use this RGD motif for on the coating of the biomaterials, so that the other cells, they can come and adhere to that petri dishes and the tea flask. So basically RGD protein is present in this fibronectin, and if you will coat the fibronectin onto the surface of the any plastic material, so if you put any cells there, they can adhere to the bottom of the plastic surface, and that's basically what we all do in the animal cell culture. And that's from here I got the insight into this into the infectivity of this virus. So what I found that this SARS coronavirus 2, it has the RGD motif that is not present in any of the previously known SARS coronavirus. And these RGD motifs, they have strong affinity. These integrins are a group of proteins that have strong affinity for the RGD motif. It means that if the virus has the RGD motif, so virus probably can take.
Hello. Yeah, actually we needed your signature and Akhilesh's signature and letterhead actually, so then I can do everything online. Okay. Yeah, that will be fine. Yeah, for this purpose we need and letterhead. So most of things I will manage online. Darpan ID, everything is I manage. I just need 3 things: 2 signatures and 1 head cover head. Yeah, so I discussed with the lady. Lady said that it's possible to take it online, everything. So yeah. That one I need 3 years. Yeah. No, these they will manage. Yeah, yeah, yeah. Okay. No, I will come back to you later in night, maybe if something is needed. First, I will try to do it myself so that it's done. And if it's not done, then I will come. I had already discussed to the lady, so she said if there is one or two files are missing, is no problem. So since I do not reply her because since we had discussion on phone. Okay, okay. See you.
The integrins may possibly be involved in interacting with the viral spike proteins via the RGD motif that is present in the virus spike protein. So, and in this way, probably the SARS coronavirus is interacting with the host cells, and it is infecting the cells and getting inside the cells and multiplying inside the cells and then coming out with many new viruses and infecting the other cells also. So I did some molecular docking experiment and I found that the spike protein of the virus, SARS coronavirus, and the integrins, they are able to bind to each other with, and I found that the spike protein, it is basically interacting with the integrins via its RGD motif. That you can clearly see here that the RGD motifs of the spike protein is interacting with the integrin. In the other figures also, I found the same thing. And but one more thing I found very interesting here is that beside the docking site, close to the docking site, in the vicinity of the docking site of the RGD and the integrin, I found calcium binding site. So that was really very interesting. It is something that maybe the virus is using the calcium ions for making the attachment or making the attachment with the host cells a bit stronger. So and the same thing what happens in case of the animal cell culture also, that when we put these cells, we have the calcium ions in the buffer, but when we use the EDTA, so it chelates the calcium ions, and then the cells they disaggregate. So I think that the same thing is happening here also. So for me, something like that, if we can chelate these bivalent ions from the lungs, in many ways, we can discourage the binding of the virus with the host cells in the lung cells, and this can be done by EDTA, which we use in the animal cell culture also, and the same thing can be used clinically also. So if we use the EDTA will chelate the bivalent ions in the lung cells, and when these calcium ions will not be present available, when these calcium ions will not be available in the lung cells, the virus would not be able to attach to the host cells. So that would be the story. So EDTA chelation, what I'm talking about, the therapy is also already known, and it is already has been practiced for the removal of chronic heavy metal or toxic metal accumulation such as lead and other. So heavy metals, minerals such as lead, mercury, copper, and many other metals can be taken out, can be removed from the body by the EDTA chelation therapy. It has been used for the coronary heart disease treatment, neurodegenerative disease treatment, for the treatment of renal insufficiency, sufficiency, and many other applications. And the most important thing is that for the EDTA chelation therapy for the treatment of the hypercalcemia is an FDA-approved treatment procedure. So if I talk about the synthetic RGD peptide-based therapy, so what is basically here is that like we have the host cell receptors, and we have the virus. So virus is using the RGD to bind to the host cell receptor. And what we can do, if we can apply the RGD peptides, so what would happen that this RGD peptide would go and bind to the integrins, and it would not give a place to the virus to to bind to the integrins. And if the virus would not be able to bind to the integrins, it would not be able to infect the hosts. And that's basically the main basis of my hypothesis and my theory. So we can synthesize this, we can chemically synthesize this RGD based peptides. And if the peptides, they are inactive, we can conjugate with some other compounds also. And a similar type of therapies related with the RGD peptide based therapy has already been used for the treatment of cancer also. And the same thing we can apply for the treatment of COVID-19 also, where the peptides, the RGD, synthetic RGD peptides are expected to bind to the integrins, and in this way, it will restrict the viral spike protein, binding of the viral spike protein to the integrins. That is basically my hypothesis. So this was the EDT chelation therapy I already discussed with you, and this is the proposed therapy related with the synthetic RGD peptides. So we have the RGD peptide. We can administer this one with the IV injections or using the nebulizer or inhaler, or it can be taken as a suitable drug delivery system also. So in one case, if you see above, we have the RGD. It is of the spike protein. It has the RGD, and with the help of this RGD, the spike protein or the virus is able to interact with the receptor. So the novel coronavirus is attached with the integrins. So, but what happens if we use the synthetic RGD peptides? If you use the synthetic RGD peptide, this synthetic RGD peptide, it will go and bind to the receptors. In that case, the virus would not be able to bind to the integrins, and in this way, it would not be able to infect the host cells. So this is basically my therapy. So there are some additional benefits with the therapy that it is related to the EDT chelation therapy, that virus use the divalent ions for the integration of its membrane, also membrane envelope and everything. So if we delete the divalent ions with the help of the EDTA will disintegrate the virus as it is also. And it has also been shown that the EDTA, it inhibits the excessive immune response generation, and it also inhibits the production of cytokines. And in case of COVID-19, the main problem is the unleashed cytokine storm. That is basically infecting the other systems, other organs also, and causing the multi-organ failure. So if we can somehow control this cytokine storm to much extent, we can control the disease also. And this EDTA, it has an antioxidant, anti-inflammatory, and immunosuppressive effect also. And that is really very important for the aged and immunocompromised people. And this technique, this therapy related with the EDT chelation therapy for the treatment of COVID-19 can not only be effective for the SARS-CoV-2, but I have a feeling that this therapy can be used for the other coronavirus also, or the future coronaviruses also. But of course, there are some potential side effects. But the thing is that the side effects, the merits or the advantage of this therapy is more important than the very, very subtle side effects it is associated with. So finally, I want to conclude here that the nebulizer or inhaler-based EDT chelation therapy and the synthetic RGD peptide-based therapy present a novel way to treatment novel coronavirus, SARS-CoV-2 infection. This technique is technically simple, quick, safe, affordable, and efficient solution for the treatment of COVID-19 patients. My work is under review in a peer-reviewed journal, and I'm also in process of developing and establishing a proof of concept and a working prototype model for this therapy. So this research has also been published in some newspaper also. And my research work on coronavirus, basically this RGD research was recognized among the top 200 researchers in India by the Ministry of Human Resource Development and ICGEB. And I would like to thank to Professor Pankaj Goel and Professor Sunil Purohit, General Secretary of Society for Promotion of Science Education Research, Udaipur, and the other professors who gave me technical guidance in this project. And I would be happy to take any questions here, please. Dr. Abhishek, if there is any question, I can take the questions.
Thank you, Dr. Tikam, for a nice talk and various things your laboratory is doing at the moment, and we are open for questions. Sir, could you tell me the position number of motif in spike 2 protein? Some position, amino acid position.
It's 402 to 405.
Okay. So, yeah, that was the question actually. Very simple question.
Okay.
So, basically, there are several methods. You can go in internet and check it. Wherever the amino acid number is marked, those sequence website you go, and then RPD motif you can check. Sir, ordinary inhalers can support?
Ordinary inhaler, yes, of course. This is very good question also, because I'm in a discussion with many doctors also, and of course this ordinary inhaler would not be able to work, or nebulizer would not. In fact, I have seen that the nebulizer is contraindicated in case of COVID-19. But yes, of course, some companies or some researchers in London, they have developed an inhaler-based therapy for COVID-19. Yes, of course, inhaler can be used, but yes, of course, these inhalers are of different type, and so that they can take the drug inside the lungs. So it is very important. So, but whatever we are taking, it should go into the lungs also. That is very important.
Then there is a question about how about ETDA interaction with other cell component when inhaled?
Yes, there are many things. There are many things about this question comes whenever I give a talk anywhere. This question comes. So the main thing is that yes, of course, the ETDA is going and it's going there and it's chelating the calcium ion there. It is lowering the calcium ion concentration in the lungs. Yes, this could be dangerous to some extent to the lung cells and other cells also. But the thing is that the advantage of this technique is more than the disadvantage of this technique. We are saving the life of any patient. So even if a patient goes down in terms of his calcium concentration or other concentration, it is okay. He would not die. But of course, the coronavirus is may die. He or she may die. Yeah, Dr. Abhishek.
Yeah. So these are the till what stage this treatment can help? There is another question like that.
To what?
Till what state this can help? That was.
The thing is that this therapy,
I will come back.
In my original publication, I have mentioned that we can take the ETTA via the IV injection also. We can take as an inhaler, or nebulizer, or through the nasal spray also. That is basically the concept. And for me is that if we are taking the ETTA and ETTA is going there and it is taking out all the stimulating all the calcium ion, the virus would not be able to interact with the host cells. The virus would not be able to attach to the host cells. So if you are at the first stage of the infection, that in that case also it will be helping you fighting against the virus. And when the virus is going inside the host cells and it is coming out with many new viruses, and that all the new viruses will go and infect the other cells. At that case or in that time also, this ETT chelation therapy would be helpful.
Can cytokine storms can be prevented by some extent?
Yes, of course. Yes, I already mentioned in my presentation that this EDTA, it inhibits the production of cytokines. It is already known in literature also. There are plenty of literature related with the use of EDTA. So, and EDTA has an effect to inhibit the production of cytokines also, and this would be really helpful in case of COVID patient. because if we are able to somehow control the secretion of cytokines, so we can prevent at least the multiple organ failure in that case.
Okay. So if there's no question, yeah, there is a question from no question but comment from Prashant. IL-17 IL-17A is known to play important role from one study to other workings as well. What could be the functional consequence of SNPs in terms of therapy in a given current scenario?
Okay, first I am not able to understand the question in
Okay,
real
the
sense.
IL-17A is known to play important role,
Yeah.
and in the current study, and others are working as well. What
Yeah.
are the roles of single nucleotide polymorphisms in terms of therapy in
No, in
current
single nucleotide polymorphism, where in IL-17, in spike protein, or in integrins, where? I think the question has something missing.
In a say a structural protein such as a spike and nucleic acid
Okay,
protein.
yes, of course. If we are talking about the spike protein, and of course, if we have the SNP in the spike protein, and this SNP is disrupting this RGD motif, in that case, the integrins would not be able to catch hold of the virus. In that case, if you mutate the virus somehow by just replace this RGD, the R from the RGD, and make this one as the alanine, we can really decrease the infection. Yes.
Okay, so that was good. Can you share the paper and slides? Meena is asking. Meena, you can go to email Dr. Tikam for a further request, and this talk is really interesting, and also Tikam had showed that from mobile phone what all you can do. I mean, I hardly do anything, but it's a mini computer in terms, and please go ahead and ask some other question if you have. We will move it to next speaker, and I think Dr. Jain is still not available, so I will take up the next talk, and I will explain about what we are doing. Thank you, Dr.
Okay.
Tikam. Thank you for your time. And
Dr. Abhishek, thank you very much, Dr. Abhishek, once again for giving me the opportunity to present here. Thank you very much.
It's our pleasure. On behalf of Institute of Bioinformatics, we are very, very thankful to you.
All the participants also, I would like to thanks to all. Thank you very much. Thank you.
Yeah. So. Okay, so somehow I am not able to let me see if I can go here, and let me see. So there is a problem. I am just trying to fix it. When I am presenting a window I want to take, and this option is not there, so maybe I have to take smaller it. Okay, now okay, first of all, like this, take a new window. Yeah, okay, good. So I'm going to start. Just a minute. Okay, are you able to see my slides? So if anybody respond by voice, it will be great because I am not able to see the screen anymore.
No, sir.
Okay, so let me see if I can see. Okay, no. Okay, so I have to go to.
Yes, we can see.
Yeah, sure. I also have to, for YouTube video, I have to go to presentation mode. Otherwise, YouTube video will come very bad quality and only audio will come and people will not like it. So now I have put it so that everything is visible in YouTube also. I check in on the machine whether I am visible. Yeah, so I am visible in YouTube. And thank you everyone. First of all, thank you everyone for coming for the in such a large number. 17 to 1800 people have been registered and another 500 have been watching without registration. And and people are from different part of the world, so that will come at the end of it. But we were working on various aspect of coronaviruses, and that will come to that. But in the research life was how was our research life in pandemic, I will explain also, and how I should be behaving with others doing that. And so we will discuss understanding about vaccines, and it's a kind of a race, and then nucleic acid protein, what we have done so far. And so I'm working at the Institute of Bioinformatics, as you are aware, and we are supported by Manipal University. And so this is a kind of a small logo from my lab, which has been now after coronaviruses jumped in, they reshaped a bit of my laboratory. And logo itself has some implications. So in general, my laboratory is working on several aspects of computational genome biology, evolutionary genomics, eukaryotic genomics. And in eukaryotic genomics, we have performed previously marine fungal genomes, insect genome sequencing, and fish genome sequencing. And just now when I joined here, we have recently received a grant on sequencing marine mollusk species, and we will be hiring a 3-year JRF on this project. And simultaneously we are working with human diseases, cancer genomics, and what are the deleterious mutation that cause a disease. Can we point out of millions of or billions of genetic variants and the cause of a disease by single mutation or set of mutations. And then at the same time we are trying to find out potential drug like compounds. And today I don't have to explain why we look for potential new drug like compounds, because everybody is searching for a drug for coronavirus. So and these are normally coming from natural products, natural product discovery basically, and characterizing natural products by their biosynthetic gene clusters. And we were working on fungal genomics and marine fungi. And after coming to India, we have started with CDRI Lucknow cooperative projects on also including lichen into it. But what is my ultimate goal of the life, or goal of my research life? So is to understand genomic diversity that is available in terms of chemical on the planet and also in terms of genetic material. So we have millions of insects, millions of fishes, millions of mutations, and how these 4 alphabet ATGC by combination permutation creates massive genetic and genomic diversity. That's all I want to focus as long as I'm alive. So with this we started working, and Corona came and it took over the stage. So a person is coming from, I came last year from Germany, and then I saw that I started setting up my lab, and then suddenly everything was vanished, yeah, because employees have to go back, everything has been shut down, and everything you are doing has to be change your strategy. So we had to change our strategy, and so we thought, okay, initially I thought, okay, I will not work on coronavirus. So what people are working, I'll focus on my area. But at our institute, Dr. Muniz convinced me, and that there is a good you see the system. And so we are working with larger genomes, and for once I start seeing about coronaviruses, then I start digging into all viruses, and then I realized, oh yeah, that is good system to work on. Although I had some idea previously, but not that extensively what I gained in last 3-4 months. So let me explain you what are the viruses. And viruses are actually non-living and living in between, something that has a minimal architecture, and it needs another species. And this is the viral zone picture where they show the sizes of the viruses. So if I want to take a coronavirus, it is 120 nanometer in size, and they are in between viruses. So the viruses can be as small as 20 and as big as 316 nanometers. So basically you and I have to, an even bigger Ebola virus. So they are of different kinds, and they will be posing a different at the moment I cannot take a questions. So the viruses will be very interesting organism to look upon, and coronavirus has provided opportunity and as well as challenge, because I could not sleep many nights because of coronavirus. So what will happen to the world? What will happen? It challenged me as a biologist. Yeah, it challenged me, one who has studied biology whole life. This is biggest challenge of our life in last 100 years. Yeah. So how that all started actually. So Wuhan seafood market, wild life market in Wuhan, they had there in the case they sell lot of organism, and it's primarily not very different from what we see in our wet markets or our markets of where we sell fishes and all. But there they have a unique tendency. They sell lot of snakes, other mammals, bats, and these are wild animals actually. So if you tap down properly and watch this lot of wildlife stuff, that actually Chinese market is consuming more pork than Europe today, and they have so much money that they buy they are capable of buying entire pork market of Europe and America, and also they are even not leaving out wildlife. So they have craze for eating, and they have become very rich suddenly. So basically what is these animals are not sold for cheaper price. They are sold for many of these animals are currently not available. Not available in China, and because they are consumed, not available in India because smuggled and sold in China. So they go as far as Africa and Latin America to collect it. So just imagine how much effort they put and how much money is involved there. So it's and then those animals lives in a different wild condition, poses a different viral repository, even probably bacterial repository, and from there the SARS-CoV-2 emerged. Okay, so there are articles, say I am just citing 2012 Journal of Virology, but if you track that on, you will see that 2003 and 2005, after it is clear that there are 7 type of coronaviruses, and coronaviruses have tendency to jump from bat to other mammals and to human, and also to bats to birds, and birds to other mammals as well. So there are 4 classes, and everybody knows that today: alpha, beta, gamma, delta, and 3 of them. So one which in infecting human are alpha and beta, and alpha and beta where there were 3 which were very deadly: SARS, MERS, and SARS-CoV-2, which is recent pandemic. And So there is a question why we had not focused that, because if I am seeing today, I would think that in next 7-8 years we have another risk, because you see so many intermediate species are there, and we are grazing jungles into cities, and then our jungles to city distances are so thin line that any wild animal can come to your house any night. And this leads into a problem that we are in a problematic zone where we are engaging ourselves with the wild a lot, and that has to be tracked down, and also the avoid our direct contacts, plus also such market sales should be also regulated by the country. Every country is regulated by country specific laws, and like we will not sell it and you will also not sell it, so every country should be aware of these laws, and what to sell in the public market and what not to sell in the and what not to sell in the market. So that's the main message from here. But then the pandemic has hit us. So genome was published, and obviously people have explained genome. You already are aware that is this 39.8 kb genome has 14 genes and encoding for 27 peptides, and so the largest peptide or largest gene is in the 5' end, which is harboring 67% of the genome, and that initially has a large peptide, and that peptide will break down into 15-16 depending on which species we are looking on, 15-16 non-structural peptide which will be chopped out and become one individual peptides itself. And then there are a structural spike, envelope and membrane and nucleic acid peptide at towards the 3' end of the genome. And when I looked at, we just deal with biosynthetic gene clusters in fungi or other microorganisms. This is one cluster. This entire genome is one cluster. So this is very minimal, and so therefore the viruses requires other host components. And the bottom slide is showing how the genome is evolved, and obviously you've seen evolutionary slides, and so basically what is shown there, that there are 4 groups, and the group wise classification is there at the bottom. So but how this first for those who do not understand how this protein is broken up, so the say ORF1AB is producing 7096 amino acid large protein, and that protein will be chopped into a small pieces, say 100 to 180 for first non-structural protein, and then second one from there into 818, and it will go on and it will produce 15 such peptides and proteins actually. So that is how it is working. And the other these non-structural proteins functions are correlated. These are the functions, but many of these functions are a speculation, or some of the functions are known, but largely they are unknown, and therefore we don't know how this virus will behave. If there is a one like Prash, as we told you on the first day, that one of these peptide is missing in SARS-CoV-2 in comparison to SARS-CoV-1. So basically viruses can, since we don't know their entire function, viruses can trick us in much better way, and therefore we have to tap each and individual component of the genome of this virus. And so unless we are knowing unknowns, so if somebody is coming at your door, you will be suspicious who is the person actually. And so same thing, if you don't know viruses so well, how we will tackle the next one. So that will be the challenge, not just for the pandemic. Maybe virus mutates and goes out of tired of infecting us and goes out, but that should not be the end of the world. World has to go ahead and tap them down. So all whatever is not known fully, we have to work on, and we should not leave our work just because the virus. Last virus was SARS-CoV-1 or SARS virus or Merck virus infected few people, and there some had thousands were dead, but then nobody cared about it, and because the literature was clearly showing that work was not done. So when I started tracking literature, there's a large gap in updates. And that is only also for scientists, and scientific thinkers also have to think that virology must go on, should not stop. And so there have been a lot of people have discussed that and have already told that it's come from bat, viruses come from bat, intermediate species, and then go to the human, and then pangolin or bat are supposed to be the virus which is infecting a new virus. So that is a question we are not I'm not going to dig in, but this is also a question to think which all mammals can be can carry. So but then when you look at the SARS-CoV, so this is coronavirus, and coronavirus is name is coming from corona in is equivalent to crown. So the spike proteins forms a crown, and therefore it's called coronavirus. And coronavirus, again, this is the architecture. The S2 receptor on human surface will bind to spike protein and other receptors as well. And then there are proteins which are structural proteins on the membrane surface, primarily envelope protein, membrane protein, and then nucleocapsid protein is wrangling around single strand DNA, and then non-structured proteins are inside. And then Parker and Smith has the paper, and then this is their summary that how to prioritize studies on drug discovery. So many people have discussed. So you have a large set of compound library, and you look for which compounds are binding or binding to which repurposed drug or compounds are binding to your protein, and how they are binding. Try to rank them and test in the organism and put it for the clinical trial. And similarly in the drug, that was for a drug, but drug is the second line of defense. So once I'm infected, I will take a drug and try to get recovered. But what if we put a vaccine, like you get an injection, and maybe after 10 days you walk out without any problem. So the vaccines are of different types, and it's quite obvious. And this figure is partly produced by one of the students, Sri Hari, will be in the audience. And the viruses can be the vaccine can be of different type, live annotated virus. So if a small amount of virus will be you take in a mild amount, and then that too can be injected. And also it's very classically known in India that when measles and other things were happening, people will go to the infected person and try to infect them manually or semi-manually themselves, so that they are not getting infected in future. So that was initial vaccination, and then you have a whole inactivated version. Then you put the small portion of DNA into bacterial vector or a viral vector or a protein subunit. Raja has already told you. And then there are nucleic acid, be it RNA or DNA virus, and then the synthetic peptides. You try to dig peptides from the viral genome and take one stretch which you can synthesize in a laboratory, and that can be given as a drug, and as an antigen, and an antibody will be produced against that. And then once it is produced, the next time our immune system will remember that when that guy comes, you have to that cocktail of antibodies. So So in the vaccine development, there are different stages, and probably you are hearing a lot, but I am trying to tell you that there is a preclinical stage. At the preclinical stage, you test on animal model like mouse or monkeys, and then you try to see how safe it is, how much dose you can give, what are the, even if it is safe, do it has a slight side effect or side effect on some, maybe skin disorder, some maybe there is sneezing more, but they are better than before. And that level, pre-clinical level, lot of test, lot of strategies will come, and 70% of that will go to the phase 1, where in phase 1 you will test on a small number of human volunteers for dosage and test efficiency, and then you extend it to say initially for 8, and then say 30, 40, 50, and to an extended phase 2, and then you go for large number of say 1000, 2000 volunteers, and you have a phase 3 trial, and you do, and then once you have done that, you will bring it to regulatory authorities, say FDA, which is one of the US one, which is claimed to be global, but you can bring it to other agencies as well. In India, ICMR is taking care, and World Health Organization, and people have question about World Health Organization issues. So probably when coronavirus or in pandemic, I would recommend not to go individually one by one, but have a global expert team who can decide, formulate a regulatory procedure, and once it is approved by regulatory approval, it will go into production and mass vaccination. So that's all overall stages are. And so what happens in normally it takes 48 say 4 years to 10 years, and but in pandemic time we have to squeeze the phase 1, phase 2 and phase 3 trials in a way that we get in a shorter period, and therefore you hear that Poonawalla has already started taking risk. If you in some month a vaccine comes out, and he is not an idiot who is doing it. He knows that vaccine is actually capable of treating against other viruses as well, can be used. So therefore he has deliberately chosen that vaccine, because he knows that either this or that he will be able to use it, and in that, if he is able to do some charity, that's really good. At this moment we do need, and we may believe that one vaccine will come, and I request you, the vaccine comes, and vaccine is expensive. If I can treat myself, I should pay for one extra. Suppose I get it in 500 rupees, I should pay 1000, and one extra person each of us will take care. Will be a good idea, and that's kind of initiative we should keep in mind. So what is going on here is that at the moment, if you track coronavirus vaccines at World Health Organization and tracker at New York Times, you will see that 125, 126 are in preclinical stages, and 7 was in the testing phase 1 on 14th June and 24th June. 10 were in the phase 1, and phase 2 has 7 on 14th, 8 on, but the phase 3 has 2 for a long time, and then suddenly one more companies yesterday came in and to phase 3, but none of them are at the final stage, and therefore you needed to so these are the 3 companies which are in the stage 1. AstraZeneca and Oxford, and has used surface protein subunit and put it into adenovirus, and then that was the first one which came, and the second one is from Australian Research Organization, which uses a BCG vaccine as a repurpose vaccine, and the third one which recently came whole inactivated from Wuhan Sinopharm and Wuhan Institute of Biological. Yesterday they came up. We'll show that one, but what is important here? So what is important here that we also understand, you are listening about R naught. What is R naught actually? So I try to explain in a very simplified way what is R naught, and many of you are probably aware. So sorry, it's dragging up and down. So already aware. So yes, as a human, I was walking on the street. A bat attacked me, and bat has virus. So I will tell you the true story. In Bangalore, in front of my house, in my balcony, virus was having a house for over 4 months, from October, November, January, February. So, and that guy will come all the time in the evening at 7 o'clock. Doesn't matter what is happening, and stay overnight. Will have as a guest stay at overnight. So that suppose that virus is carrying a coronavirus, and I get infected. Once I get infected, if I will transmit to one person, then out of 5 when I meet, it's a 20% attach rate, and how many contacts I come into play, that will decide an R naught of 1. And then when I started infecting more than one person, the R naught will increase to as start infecting more people. So if R naught is very high, that we have to worry about, and if R naught is going to 1, or if lower, probably, then lower it is, better it is. And so what we are also hearing about herd immunity is good, and this that. So herd immunity is good when there is a vaccine, or people are recovering and have immunity. So initially you have a novel outbreak, and then the population has few are infected, no immunity. Then some get immunity because of recovery, or because of vaccine being available, and then that prolongs for a while, and people get immune to it by virtue of vaccination plus recovery. So for that also vaccine needed to be required, and it may work in some of the less populated country, and but we are in a very highly populated country. We should not expect to wait for herd immunity, because in a herd immunity also is successful only after 60 to 70% of population is infected, and that you do not want to go, because if the herd immunity will not come, you will be left out with very less human resources. A human population, our population is bad also, but population is our strength also. So we can't ignore that, and we do not want to lose anyone. Every life is important. And so there are questions time to time coming to us, why so many vaccine attempts are there. So these slides is making clear, there are only 3 of them are actually at phase 3, and maybe one of this will hit to the target. Okay, so that's why it is important that we are having a different strategy, and everybody is trying, and that's really good. And that's what I said, the Chinese dailies report that Chinese vaccine is available, and they will test it in UAE. I'm not sure why they will not test it in China, but that's their strategy. So last night that was in news. And so now I come back to what we were doing. We were trying to characterize nucleic acid protein and coronavirus, and then we team up with everybody who were available to us. And pandemic had hit. I could not wait that my team comes back and we start working. So we team up, and the initial idea was from Munish, Professor Munish, Dr. Munish for institute, and he has initial ideas, and then we can go ahead and pull it out. And then this paper is currently under review in Nature Microbiology. Preprint is available in the preprint.org, and you have if you want to have a look, you can just search the name. So what is known about what we try to do is try to characterize a nucleocapsid protein from various angles, and it can, what we saw initially by computational analysis, it can go to the nucleus. It has a RNA binding function, sure, because it is circulating the RNA, and then it shows that it is a extracellular, intracellular, cellular compartment impact also, and that both nucleus and cellular component, it gives us an idea what the hell is happening. It's in the nucleus also and cellular compartment also. What is going there? Some sort of a hint, and I will come to that. So nucleocapsid protein protein is, as I told you, as it at the 3' end of genome of coronavirus from Wuhan at the bottom, and then we compared with the Indian samples, and also we compared with the using UCSC Genome browser. We compared that one with the 43 bat coronaviruses, and we see the conservations, and but this is same position is occupied, and it was obvious also, but we had tested that, and then when we examine further, what we see that this interestingly, this protein is 419 amino acid long. It has an unstructured region in the 3' end and 5' end, and then RNA binding RNA binding N-terminal domain, and RNA binding and dimerization domain C-terminal domain. These are, and then the linker domains, which linker region between the two main domains. What we identified is the 7 motif that could be useful. And also we found that nuclear localization signal, nuclear export signal, both are present. So 2 nuclear localization signal, 3 nuclear export signal, and that will show you some. And then poly Q region or Q rich region also we have identified. So what we see here is the alignment, simple alignment, and we have taken this closed species, and we have taken some of the coronaviridae far species which are away as a representative sample. And what we see here that there is a motif 1 which is highly conserved, and then motif 2 is highly conserved, and we keep on looking, and then what we see that there is a nuclear export signal is present, and then we have second nuclear export signal is present at the motif 4, and motif 5 is present, which is also a nuclear localization signal, and then motif 6 and motif 7. Motif 7. But what is there? What we additionally found that lot of glutamines are present, and glutamines are Q, denoted by Q, and there is one strong patch, and then several conserved patches, and which are marked with a star, and the stars are in which species they are conserved, not conserved. Those positions are marked. So in SARS-CoV-2 we find one of these Q is not conserved, which is conserved in all other viruses, and so the kind of a stretch of glutamine rich region, or patch 1 we named it, and then in the second patch is present at the end of the C terminal end of the protein, and we'll come back to that. So what we saw that you all are also aware that SARS-CoV-2 is close to bat coronavirus 2, even closer than SARS-CoV-1. So is it somebody writing? So then what happens is that what happens is that these are the motifs, 7 motifs we have found reported, and these motifs are localized on a structure as well, And then additionally, when we check this motif, so I told you this motif is conserved. Now what I'm showing you, that is not just only conserved for SARS coronavirus, but also the 4 classes of the other members of the coronavirus viridae. And then that is a highly conserved, one of the highly conserved region in entire genome. And that shows that if we target this as antigen, probably that will be able to suffice in controlling all pathogenic coronaviruses. And this has to be tested further, but then this is so conserved that it has not mutated until now, when probably next mutation is also in a very ultra conserved region, although we don't know full function of it, and we will be testing that further. And then what we saw, that second motif, which was coronaviridae specific, motif 4, and that was also nuclear localization signal, but that was only present in SARS coronaviruses, and the close relatives in the bat. So this is a nuclear localization signal. We computed nuclear localization signal in SARS coronavirus viruses, plus then we have also computed these SARS nuclear localization signal in the known nuclear localization signals, such as in HIV and also in human limb kinases. And so that shows this is a nuclear localization signal, plus this could be also can be a targetable region. Then what we saw, that as I told you, this is disordered or unstructured region. Unstructured region means amino acid composition is repeating again and again. So poly SR or poly Q region, and then when it is more than 0.5, that's when it's one type of residue is repeating. Otherwise, ordered region will have amino acid at a random frequency depending on what codon it is. But if the codon is constantly, more or less constantly producing one amino acid, that becomes disorder region, because that will lead into non-folding and have a not a classical definition of protein. And that's what we have saw, and that was present in the, not just in the SARS coronavirus, but also in the other corona members of coronaviridae, and that's more or less constant, and we have tested with the different tools. And then when we saw that glutamine rich patches, with the glutamine rich patches were present in the extensive in number. So initial one is 20 amino acid, 20 positions have glutamine, and positions are marked with the Q229, means is present in SARS-CoV-2. But if the SARS-CoV-2 has something else, say A267, means SARS-CoV-2 has A and Q is present in other viruses, and so it's actually numbering is according to the SARS-CoV-2. And so these are first Q patches are 20 and second is 16. And then what we correlated with from the previously known literature, and also known in literature specific for RNA viruses, that these two nuclear localization signal and nuclear export signal will actually help dynamic transport, that nucleocapsid protein will go into the nucleus of host and come back, and probably it's whatever it attached to it will also go back and come back. So as a dynamic transporter, and dynamic transporters are more and more getting clear by harboring these two motifs in other eukaryotic systems, in viruses as well as eukaryotic systems also. This is already getting well established, and then these motifs are aggregate forming. Our study also found that these patches plus motif 4 can form aggregates, and that so they are a dual functionality, and it's already known that nucleocapsid protein has multiple roles. it's not as striking as spike protein, but it's one should not forget it. It's a kind of a forgotten hero, and therefore we have also did drug repurposing for this protein, but I'm not going into that detail. It's available in the paper, and we have found several antivirus viral drugs from FDA already. So we're not going into it, but this is the biology of understanding biology is more important in my. If you understand the system, you can actually tap it better. And so that's my idea, and obviously others want to have a repurposed drug and other things, so we have done that. But for me it was understanding the mechanism. So this mechanism, similar mechanism, is also known in some other RNA viruses, and you have to read a lot to come up with that, and hope that reviewers will agree, and if not, then we need to do tests and all, and obviously we'll also try to test it in future. So at the student level, and also for the researchers which are new, what is we are focusing on actually here. So this is envelope protein, and the two students have wrote a paper, and what I am trying to tell you here is that what we are trying to dig is we are trying to see take a beta coronavirus as a central and say SARS-CoV-2 is marked in a blue as obviously a prime object, but then have the understanding of all 4 coronaviruses types in general, and we are focusing on that we are not tapping all of them by means of comparative genomics and whatever information we can pull it out from genomic analysis, and then we'll pull it to the public, and then public so we made our publication out before it was published, so that others, if they find some angle, they can actually use it. And that's the need of the hour, and that should be need of the future as well. And so with that I will conclude, and we were having at BioClue Society we have International Journal of Computational Biology, and there we are also having a special issue on international on coronavirus genomics, bioinformatics, drug discovery, drug repurposing, population studies, machine learning method, viral informatic databases. So if you have an issue, please come up, and this slides is slightly older. I think me and a team is there, but you will find details in the International Journal of Computational Biology. But before that, I finish my talk. I would like to thank Department of Biotechnology, which is funding my Ramalinga Swami Fellowship, plus the recently received grant, and the Institute of Bioinformatics for wonderful infrastructure, and we are also receiving support from Manipal. And during COVID-19, the Spanish company BioBam or Omics Box has also come up to support my COVID-19 work by providing their computational infrastructure and also the Omics Box software. So that is very good. And then what happened in the bottom is you see me and some of the students who are currently working, and PhD students as well. These were the original team, and then during coronavirus, most of them struggled here and there, and some of them vanished, and quite obvious. So it's a pandemic, so you have to save yourself, and if you are not available, that's not a problem. 2020, I will keep that okay, because everybody survives, then you will do the research. But during pandemic, I found a lot of people come up to support the work we are doing, and this is coronavirus, COVID-19 volunteers, and volunteers are from some of them are, say, Gaurang is from Sweden, Srihari is from Bangalore, and from every different parts of the India, and some of them are shy to keep the photos, so they're smiley smileys are there. So and then Dr. Mitra is a very senior scientist, and he's originally from chemistry, but he's taking up epidemiology studies along with us, and he has a PhD from same university where I did my PhD, so that time I know. And he come up with that they're doing from chemistry and has lot of good research in chemistry, but he came and partner up at the time. And Dr. Parveen has machine learning expert, and they all are participating to good length, and I have only given names of those volunteers who have participated. Many of them could not participate, so they were shown intent but not. And then these are collaboration, these are the yellow ones are Dr. Jyoti, who internal in IOB, Dr. Munish and Dr. Anusha, and have been worked together. And Munish and Jyoti, you saw in the paper, Dr. Anusha is working with the vaccine strategies, which we are thinking what we are thinking that she's reading, and we are writing about that. And then Dr. Tikam also, we got in COVID-19 and get in touch with due to COVID-19 work. And Dr. Pankaj, Dr. Sunil from Dr. Pankaj from Suraj, and Dr. Sunil and Dr. Ravi Tandon from JNU. We wrote several grants during this period on COVID-19, and until now none of them are actually fully done. So, but we will keep on working. So that's more important is that you work, and work will obviously sometime you will be rewarded for that. And but the team has so what I'm saying is that we extensively working, and most of people are working from home. So most of the volunteers, we are currently not able to pay them. We had no budget for that, but they showed a great courage. And one of these volunteers initially started as a volunteer, Tejaswani, jumped in and become member, and now she's a PhD student, primarily focusing on coronavirus. And so we will take care that needs of these tough time co-workers, that they get suitable positions or whatever they want to do. We will be supporting them. And so if you want to become part of our team, please contact us. You can look in our advertisement in India Biosciences, GitHub, BioArchives, Job Archive, and ResearchGate. And obviously you can always go to our institute's website, ibioinformatics.org. My email ID is there, abhishek@ibioinformatics.org. Mobile number is also there, but do not call me at 2 a.m. in night. I may be awake, but it will sound a little bit but if you have a question in daytime, you can reach me. With this, we will end up. And yeah, so the 2 smiley faces, and we probably have money for 2-3 people to join, and we are constantly looking for applicants. But do not send just CV. Just look in GitHub or India Biosciences or BioArchive what kind of suggestion I have given before you apply. So there is a guideline what to write in email. And if you are not writing in email, then I am not sure what you want to do. Just by CV, I can't make any judgment. So with this, I will hang up, and I thank you all for staying with us for so long. And some of things are new for many people, don't know who are working with us, but today clear to many of them. And then obviously they have credit, so they deserve to be on the credit slide, and they will be part of our future publication. So so with this, I will end up, and I will be open for question. But before that, I will ask whether Dr. Jan has arrived.
I have.
Yeah, okay, good. So after me, you will be presenting. And so please, I'm open for question. Guys, please ask me if you have one. In the chat box is not clear to me. So if Prash is there, can you moderate it for a minute? I can see now I can see the entire webinar. It was wonderful. Okay, thank you, Gogoi. I can now see. Okay. So no problem. I can see that. With this, Madam Urmila Kulkarni College writing good work. We are thankful. We are learning from you, Madam. And the Legume Lab is saying, do we have any master program? We don't have master program, but due to COVID-19, everybody is working from online, most of them. If a student is really interested and can pull out their master work or 8 on say 6 to 1 year work from home, we are ready to train them. We provide them good training, and many of participants are or COVID-19 were master students also, and out of choice also people came. So if they are very much interested, and in 5 days I will be able to track whether they will be able to come back or not, and it's because then we can actually help them without any in COVID-19. We do agree that many master students will have a problem, so we will help them, and because the laboratories many experiment one cannot do, so we'll help them and to their best capability. So we'll gauge their capacity by just talking to them, asking some questions, and based on that we'll design some project so that this period will go. Drug target using drug targets are available in your paper, so I do not go into detail, but you can read in the preprint. So we have scanned entire drugbank.canada, and that one is already present. Sir, eligibility to join your lab in our institute. Dr. Jan is probably not aware, but we follow his papers very seriously on nanopore. So last 3-4 years he has been remarkably publishing on different aspects of nanopore. Say one paper on human genome assembly improvisation, I think is Nature Biotechnology 2016, if I am not wrong. And we were knowing that someday we are going to meet, and this is the opportunity. And thanks I thanks Max from UCSC who came yesterday, who introduced him to us, and we were very glad that he could make it, and he's one of the brightest person in nanopore or second generation or third generation sequencing. So please stage is yours, sir. And in India we call sir, so that thing is I'm getting back. But sir is a good way also to start appreciation. So I was in Germany, and my boss will ask me to not to say professor, and in Germany it's compulsory to say professor. And I will honor. So I one day explained him that I can call you by name, but I honor your work. So that work and the first you put, so the word professor, let me keep it. And so your work makes you to be a sir here. And please go ahead. And I am stopped. Oh, I stopped. Rejoin.
All right.
I was somehow disconnected. Okay, I pressed wrong button. So
what I'll talk to you about is a little bit of not a little bit, a fair bit of background with nanopore sequencing and what it can do, what it is, and then how it has over the last few years been applied to pandemics and outbreaks. And of course, there's a lot of aspects to any sequencing technology or genomics in particular. You can talk anything from E. coli or bacterial genomes to human genomes to plant genomes, and everything has a different application. In this context right now, I want to give you an idea of what the technology is able to do and how, using a couple of examples, has it been applied to outbreaks and pandemics around the globe. What you see on the image on the left is a MinION, and on the background here is a cell phone. And I'll tell you later which cell phone it is. You can kind of make a guess calculated on that. It's a very small size portable device. And then on the right is its Hulkbuster cousin, if so to speak. If this thing on the right is a laser printer sized device that's essentially able to meet and beat Illumina NovaSeq levels. So you're essentially talking about multiple terabases of sequencing data per day. So this is an iconic picture from the Second World War, and this has some of the finest scientists of that era, Albert Einstein, Richard Feynman, etc. And just to give you an idea, I might talk about some of the things that may be very obvious to you, but sometimes it's easier to approach a conversation thinking that the smartest person in the room knows nothing about what we do. So to give you an idea of how complex biology is, if you take every living thing on Earth and viruses, not to leave them alone, if we count each of their genomes and ask the question, how much nucleic acid is in our biosphere? So this is every kind of living organism, every kind of virus, every living form in some shape and form that's alive or has ability to continue proliferating. That number is quite large. Turns out it's on the order of 10 to the 37 bases. And the idea to give you is on the right is a supercomputer. It's called the Summit supercomputer at Oak Ridge National Lab here in the US in Tennessee. And the basic idea is you take the 4 fastest and most powerful supercomputers in the world, put them into one, and then 10 to the 22 such computers will give you enough capacity to store these data. We are not even talking about compute. This is purely storage. So biology is complex. And as much as we understand, we have literally not even scraped the surface of the genomics and science that goes in this nucleic acid space with sequencing and genomics. So the reason we do nanopore sequencing a fair bit is, one, I work from a nanopore group. I did my PhD at this university as well. And the other reason is nanopore sequencing was invented at UC Santa Cruz. Dave Deemer, who's a professor emeritus in our department, was the original inventor and pioneer of nanopore sequencing. The idea was conceived in 1989, along with George Church and Dan Branton, both at Harvard University. The person on the right, Mark Akison, who's my mentor, he is one of the pioneers who has been working with nanopore sequencing since 1996 and has made a lot of strides in terms of how the technology works and what it was able to do. The drawing that you see in this is a very iconic drawing, literally one of those things where a picture is absolutely worth $1 million in this case. Dave Deemer was driving on a highway. He had an idea. He pulled over on the side, took out a piece of paper, drew this scribble, and this is literally what went into the original nanopore patent as the idea. Why do we call nanopore sequencing a breakthrough technology? It has a lot of features that are somewhat unique to it. I won't go through the verbiage here. It's just to give you an idea as I go forward in why we think what it is. It's a single molecule sequencing platform, which is to say that any molecule that's sequenced on it is being sequenced one by one. There's no sequencing by synthesis, so it's not based on copying DNA or RNA. It directly sequences native bases. So it's sequencing genomic DNA or genomic RNA. There's no amplification required, which makes it very cool because, as I'll show you later, it can see methylation marks, for example, or other modifications. To this date, we have not come across, and this is not to say it can't be possible, but as of now, we haven't come across a base modification or a sugar modification that we could not see in the nanopore data signal-wise. It's there. And there's no theoretical limit to how long of a strand can be processed. And I'll show you what that means. But the longest DNA sequencing read that has been done to date is 2.4 million bases. Compared to, on average, when we look at Illumina data, we are used to looking at 150 bases long reads. 2.4 million bases is a very big paradigm shift. How does the technology work? So this is a very, in a nutshell, it's a very simple concept. Think about a lipid membrane that's in all our cells. So think of a membrane that's synthetic. This membrane is separating 2 salt solutions. Now, it's impervious to anything. When you insert a protein nanopore in this membrane, what you have is essentially a hole, something like this. So you have a membrane, you have the pore, this is a hole. The hole is separating two salt solutions. When you apply potential across those two chambers, what will happen is ionic current will flow. So you have a flow of ions going through that hole. When a strand of DNA or RNA transits through that hole, it will interrupt the flow of ions going through that hole. And that flow of ions that get interrupted can be detected as changes in ionic current, as you're kind of seeing these ups and downs squiggles here. Those ionic current changes can be used with deep learning methods to infer the identity of what went through. So once you start seeing signatures that this kind of signature may be associated with some kind of bases and so on and so forth, it gives you the ability to process native strands and then discern the identity of those strands using the ionic current. One of the hallmarks for sequencing technologies is that what can you do for genome assemblies? Generally in genomics, you're looking at two main paradigms. One is reference-guided analysis, which is doing a lot of alignment to a reference and then inferences based on those alignments. The parallel route is how properly can you make a new genome. So if you did not know anything about some data, but you knew that it represents a genome, can you make good quality genome assemblies? That's one of the yardsticks for things. And in that, I'll also tell you where the nanopore technology at the moment stands. So if we look at the performance today, the technology has a median identity, or in the more conventional terms, sequence accuracy of around 95%. And as I mentioned before, the read lengths are exceeding 2 megabases. A hallmark of technologies is human genomes. It's just a yardstick that is kind of measured with it. And as Abhishek mentioned, as an international group of collaborators, we were able to assemble the first nanopore-based human genome back in 2018. And at the time when this assembly came out, it was the most contiguous assembly done using a single platform until that time. This has been surpassed since then. We have done a single chromosome into a single assembly. We have done a centromere at this point. We've done recently a new manuscript that has come out where we did 11 human genomes in 9 days. And of course, there's always scalability with these things. You have to devise new technologies and so on and so forth to handle the data. We're getting to a point where terabytes of data is not a problem. Handling the data ends up being an issue, and managing it because of the obvious reasons that one might imagine. Too many things, too large volumes, and that's where things can go wrong, and at times they do. 2.4 million bases is the longest nanopore read till date. And as I said, conventionally, we as a community are used to looking at shorter reads, at best on the order of a kilobase or few kilobases. So to give you an idea of what 2.4 megabases means is to give you an idea, to give you something to imagine. This nanopore, if this nanopore on the left were your hand, so if you close your fist and that was the scale of the nanopore, which instead of 120 angstroms, now we are talking on the order of centimeters, then a 2.4 million base long read would be something on the order of 7.5 kilometers long rope. So now think about your hand and think about a 7.5 kilometer long rope going through your hand. That is the scale of processing that the technology can do. And at the moment, the jury is out, but at the moment, we haven't come across a limiting factor in this being the technology. Rather, the limiting factor is the integrity of the DNA that goes into the system. It's very hard to handle megabase long DNA molecules, which makes it very hard to capture it on the nanopore. But if you can deliver it to the nanopore, you will get a sequence out for whatever length of molecule that you deliver. And this applies to RNA. Of course, we don't have millions of bases long processed RNAs, but the longest sequenced RNA strand to date that I've seen is about 31,000 bases long, which in itself is a marvelous thing when you think about a genomic RNA strand isolated from a cell that's 31,000 bases long and was read end to end. Some of these are transcripts that contain as many as hundreds of exons in a single go on a single read. One aspect of nanopore technology is that it's so portable, and we'll come to that it allows you to democratize sequencing. Part of this is that ultimately a technology, or most technologies, aim to get into point-of-care clinics, in hospitals, in different clinics, and think about a world which we will see in likelihood in the next few decades, if not sooner, is precision medicine. when we go to a doctor, anybody who needs some treatment is not going to get a boilerplate kind of treatment that here, take some drugs and go. What might happen and what will happen over time is it'll be customized given your genome, what you need, what the composition is, and then how can it be applied. So one of the things that Nanopore does, because of the long reads, it allows you to phase reads and do better structural variation analysis. So what phasing here means is that we all have two different alleles. I got some part of my genome from my mom, some part of my genome from my dad. And with long reads, we can go through enough of a stretch of nucleic acids in the genome that we can tell that I see variation X in this part of the reads and not in that part of the read. So I can tell that a set of reads here, which have the variation, come from one allele, maybe could be mom or dad, and the other set of the reads that does not have this variation must come from the other allele. In addition to that, structural variation, which is generally hard to perform with shorter reads, becomes easier. The reason for that, if you have a deletion that's 10 kb long, but you have a read that's 50 kb long, it's an easy thing because you have 50 kb of alignment, and some of it is going to be distributed on either end of the 10 kb deletion. Very easy to look at. Similarly, variations in exon coding regions, etc., become easier as you look at with long reads. And the last bit, which I think is one of the coolest aspects of the technology, is direct simultaneous detection of base modifications. What that means is that, as I showed earlier, when your bases are going through the pore, they change how the ionic current flows through those holes. Now, if you have a cytosine going through from that hole, it will have a characteristic ionic current. Instead of that cytosine, if you had a 5-methyl cytosine going through the pore, it will have a slightly different ionic current profile associated with it. Here's an illustration from an E. coli 16S ribosomal RNA run. What you see as these squiggles in red and blue is kind of the representative ionic current for sequence going through the pore, like you see on the bottom. And what happens is, if you see on the left-hand side, the red and blue kind of overlap, and then on the right-hand side, the red and blue kind of also overlap. In the middle, where you see that highlighted G at around position 528, that is the region where you'll see that the red and blue deviate from each other. And why is that? It's the same sequence in both of those experiments, but in one case, in this case, the red one, the G is actually a G. In the blue case, the G has a methyl group on the 7th base in the sugar. So on the base. So what it is a 7-methyl guanosine in the blue and a conventional guanosine in the red. So you can see this is a classic example of how modifications going through the nanopore produce different enough artifacts in the signal space that you can then use to infer the identity of the modification and also analyze the modification itself. And this makes things very interesting because you can imagine doing DNA methylation studies where you look at gene regulation, same thing with RNA modifications. RNA is very cool because in DNA, we generally think of cytosine modifications, adenine modifications, maybe sometimes other aspects. In RNA, there are 150 plus modifications. So there's a lot of things that we do not know. And this is one of the tools that we think could be and will be used for that. As I mentioned before, it's a small device. It's a portable device. And the reason for that is it was designed in a way, the MinION itself is a 90-gram device. If you looked at it in a lab, it's kind of even hard to see. So just jumping to the slide, on the top left is the same image that I showed on the first slide. The phone in the background is an iPhone 5S. So that's to give you an idea that how small it is. It basically is like a stapler that's sitting on a table. It works from a USB 3, very easy to configure hardware. Standard laptop will work. There are other versions of MinIONs now that are even more self-contained. There's a new MinION device from Oxford Nanopore now that basically can do everything in think of an iPad mini type device, and it has even cell phone connectivity built into it. It's very portable. People have taken it into extreme environments, Antarctica, the Arctic areas in the world. They have taken it into the deserts in Chile. So very extreme environments, it works. And the most extreme example that I think of when in-field sequencing is talked about is the picture in the middle, which is the MinION on the International Space Station. The picture of astronaut Kate Rubins is from 3 or 4 years ago. from the first time DNA was ever sequenced in space. Think about it, it's the only technology that can actually fit onto a cargo vehicle flying off from NASA to reach the International Space Station and then actually be able to do the sequencing in space. And that's very impressive. Similarly, the picture on the far right is of astronaut Peggy Whitson on the ISS as well, and doing an experiment from 2 years ago using the MinION. The picture on the bottom left, which has the map of the world with all those shiny yellow colors, those colors or lights kind of indicate what parts of the world are MinIONs being used in. And essentially, the bottom line is that it's used very much globally. There's over 10,000 MinION devices at the moment in the world, which is unprecedented. It's the most distributed sequencing machine because there's no other technology that has 10,000 sequencing machines around the world. The idea with MinION always was to democratize sequencing. So back in 2014, when the MinION Access program, an early access alpha beta program started, the goal was that if you're a standard lab and you wanted to do some sequencing, you can buy a MinION and do it in your lab. You don't have to send your samples to a sequencing center, wait for a few months for them to fill their lane, do the sequencing and send the data back to you. You can do the library prep yourself, sequence it, and basically have the data in a matter of hours, if not a day or two. And in the context of what we are discussing today, I'll spend a little bit of time now going into how the technology in the last few years has been applied to different types of outbreaks and pandemics. And I'll quote a few examples, largely from the UK, where a couple of colleagues, Josh Quick and Nick Lohman from the University of Birmingham, did some early pioneering work with applying MinION technology to outbreaks and pandemics, vis-a-vis Ebola and Zika. In 2014, there was this news that many of you may not have seen, where at a hospital in the UK, in Birmingham, they had a salmonella outbreak. And it was very quick. Within a day and a half, it closed down 8 hospital wards. At the time, Nick Lohman and Josh Quick used MinION and along with Illumina MySeq and asked the question, can we apply a real-time sequencing technology like nanopore? Can we apply it to an outbreak like this and look at what the cause of the outbreak was? And it turned out, when we do conventional methods where you isolate DNA, do the library preparation and sequence it on the MySeq, it takes about 40 to 50 hours to get the answer, which is pretty good. For a hospital setting, that's fantastic. But they were able to do the same thing with the MinION in about half a day. So in less than 12 hours, they were able to get an idea. One thing that I did not mention early, what I mean by real-time sequencing is when the ionic current that's arising corresponding to a nucleic acid going through the core, it's real time. So I can take, for example, if I were to sequence something right now, I could even take the ionic current corresponding to the last 10 seconds, base call it using deep learning frameworks and have a sequence. So it literally, if I had a 16S sequence, for example, that we were searching for in a metagenome and asking for what the pathogenicity is, we could take the last 1 minute of data, look at it, and see if we are finding something. And we can do it repetitively. So, and because of that, what that means is that it's very real time. We don't have to wait for the experiment to finish. We can start looking at the results within seconds of when the experiment starts. Now, this salmonella outbreak was very encouraging. Encouraged with that, Nick and Josh applied the MinION to the Ebola outbreak in West Africa in 2015. And at the time, it was an interesting challenge. And what they did was they set up a mobile lab in field in Guinea. So literally the picture on the top left is kind of what they had to carry, lab in a suitcase kind of a setting. You take everything that you need, you have to think about what you might need. They had to carefully plan. They took everything, set up a lab in Guinea on that table that you see on the top right, and did real-time in field sequencing as you see on the bottom with the MinION. And it was, it has its own challenges. Internet, for example, is not the best at every place in the world. So for example, in Guinea, Josh was going into a hotel lobby to use 2G network to upload parts of data, make analyze that in the UK and make inferences. And why is that interesting? The reason is that if you can do a more real-time analysis, then what that allows you to do is draw time-scaled lineage graphs. So what that means is that if you look at this phylogeny tree, where you're trying to track different types of strains of Ebola, the simple version to look at this is that the red colored dots are principally closely associated strains. So you'll see clusters of red, as you see in the top part, and then a small cluster in the bottom part, close to November. So this can allow you to draw trends. And what that means, it'll clarify in the next section. So the last thing that we'll discuss in my brief talk today is SARS-CoV-2.
Someone, please, your voice is disturbing.
Where we will be today, and it's a strange world, and the fate largely defined by SARS-CoV-2. The good thing with COVID-19 was that the scientific community across the globe came together to address this challenge in a variety of ways. Different types of detection tests, different types of vaccine ideas, or whole genome sequencing, epidemiology, contact tracing, whatever have you. The community has amazingly worked well, and within reason, not as much sharp elbows as people may associate scientists to have. So it's very cool in a way that, as a silver lining to this pandemic, it really did bring people together, whether it's people in general or people in the scientific community. Since we are talking about sequencing, there's an element here that we can do whole genome sequencing for SARS-CoV-2 using nanopore or Illumina. The idea stays the same. You take the viral sample. Once it's positive, you do a reverse transcription. You do a PCR amplification using tiled primers. The tiled primers are to give you enough representation of the whole viral genome, which is 29 kb long, fairly large genome for a coronavirus, but for viral genomes in general. And then at that point, you can sequence those PCR amplicons with conventional MiC, or you can sequence them with nanopore sequencing. The nanopore workflow tends to be a start to finish. It's an 8-hour workflow at the moment. And this is for whole genome sequencing that allows you to do epidemiology, which Illumina can as well, with a little bit more time spent in sequencing. The question arises, why do we care about whole genome sequencing? You heard from Max yesterday, the kind of lineage trees that they are drawing with the UCSC COVID-19 genome browser. The reason epidemiology is important is that you want to know how the virus or how any pandemic causative agent is progressing in time. The example here, for example, is on the bottom part of this figure of the phylogeny tree from next strain, you'll see that it's largely dominated by blue dots. That's the Asian strain, which is principally derived from the Wuhan strain. And then you'll see over time, starting in December through January 29th, the blue spots principally dominated Asia and not really beyond that. But over time, you'll see that as Europe and North America started showing up on the map with time, you see the strains also cross over. So it's not that they've got their own version. Principally, at least until the month of March and April, the two largely dominating strains were the European strain and the Wuhan strain. And this is important in monitoring the change and evolution of the virus, because if it's going to change, if it's changing at all, or if it changes at any point during the study or during the pandemic, we want to know. What's important is that once you know that there is a pandemic, you want to also know if and when it changes, because a lot of times these viruses do. Now, here's an example. I'll just let this run in the background, where based on these information, you can also start tracking how the pandemic is spreading. So it started from China, slowly progressed to the US, there were clusters, and now there are clusters all over the world, India, Europe, North America, South America, everywhere. And this is yet another thing where epidemiology is useful, because this is the idea in a large sense, and then there is the small scale application with contact tracing, where if you know what kind of strains you're monitoring, you have whole genome sequencing data, you can look at if patient 1, the strain that they were infected with, was closely related to patient 2, who might be another person infected with the virus, and so on and so forth. If one is closer to 2 or one is closer to 3, if you can start drawing trends, and you can start looking at okay, one is closer to 2 and one is also closer to 3. So that means they might have been from the same cluster, they got the same strain, and they might be geographically close. Now, let's assume there's a patient number 4 who came from Europe. In India's case, this is quite true. We all know of stories where people came from international travel and infected other people, knowingly or unknowingly at times. And the strains were different. So that was, again, an important feature to address. By understanding the epidemiology of the virus, not only can you draw trends globally in terms of its spread, but also associate that in a local sense with contact tracing with respect to knowing the strains that you're looking for, how it's infecting and how it's spreading. One of the things that obviously we have now heard a lot, the word testing. I think saying it to anybody, I think at this point, anybody who does not know what testing in this case means is probably living under a rock for the last few months, or was in the Himalayas with no internet and newspaper access. There's a lot of ideas, a lot of companies in India as well. I did not put their names, mostly because of my own ignorance, which I apologize. And I have heard and seen news of people doing groundbreaking qPCR developments in India, as well as vaccine development. There's a lot of good ideas with testing, where you can do hundreds of tests a day at some cost and whatever have you. There's different strategies. I'll spend a minute or so here telling you those. qPCR is a standard one. You take a couple of regions of the viral genome, generally in the N protein, a couple of amplicons. If you see the amplicon, the patient's positive, the sample is positive. That's the qPCR strategy. There's LAMP-based strategies. LAMP is loop-mediated isothermal amplification. This is interesting in that it can work from low viral copy inputs, and it can give you an assay of did somebody's sample have a viral infection or not. But what's nice about it is that it does not require a PCR setup. It's essentially an isothermal amplification. So generally, you incubate your sample mixed with the mixed with the amplification reagents at 60 degrees for 30 minutes, and you have your material for sequencing. So it's really, really very quick. There's some lateral flow assay-based techniques where people will do kind of mix their sample with kind of an antibody. So think of an antibody that's done on an antibody assay that's done on a membrane, where if you see a color, it's positive. If you don't see this color stripes, it's not positive. And the idea behind that is using a combination of either antibodies or Cas9 or Cas13 kind of DNA or RNA binding agents. And the idea is the specificity of the Cas system or the specificity of the antibody is what you use. And the whole rationale behind all of this is how quickly can you make it, how fast is the test itself, how scalable it is, and how cheaply can it be done. And all of those really translate into large-scale deployment. If you do 100 tests a day or 1,000 tests a day, it's no use to anyone. US and UK are classic examples of how the absence of large-scale testing can really make a big difference to how you address the pandemic. In that light, the last thing that I'll tell you is something that Oxford Nanopore is developing, that a company that we collaborate with, and we are going to try and implement this technology at UC Santa Cruz, as are many other universities. The idea here is LAMP core. So what it's essentially is LAMP plus nanocore, and the idea here is you can do LAMP-based amplification. So very simple workflow. You take the sample, isolate the RNA, add the reagents, incubate at 55 or 60 degrees for a 30-minute time, have a 20-minute long library preparation, sequence it. And if you see the amplicon on sequence data, and again, like I said, nanopore sequencing is very real time. If you see it, the sample was positive. If you didn't, it's negative. And it's fairly sensitive. At the moment, at least we haven't tried it. You're looking at the white papers that Oxford Nanopore has shared on their website, but it's very promising. And the really promising part, in addition to all of the strategies being simple, is that the GridION, which is essentially a small kind of a mini desktop printer size device, and it's essentially the equivalent of 5 MinIONs. With a GridION, you can basically do 5,000 samples every half a day, so 10,000 samples a day. And because of the workflow being so simple, it's very automation friendly, because all you have is collect the sample, and even better if you can do it from saliva, which I believe is something that they are working on, and we'll see when the technology comes to research for us to test with. but once you have sample in a tube, it could be automated in that a robot can do the extraction, add the reagents to the tube, incubate it for a particular time, and then just prep the library. So 10,000 samples a day is not really out of reach. And the cost, at least from a scientific standpoint, because we as scientists think about how expensive it is to really deploy in large numbers. At the moment it comes, I think it's very affordable. It's much more affordable than most conventional testing kits are at the moment, at least in the US. I'd be curious about what the testing kits are in India and how the costs are for those. But it's a very promising new nanopore-based rapid diagnostic coming out. And I realize that I'm the person standing behind you and dinner, so I will stop here. There's a lot of collaborators, a lot of people whose work goes into a team. So no one person does all of the work they describe. So there's a lot of collaborators. I won't go through them name by name, but there's a bunch of them at UC Santa Cruz and then at other universities and different institutions within the US and outside. So with that, I'll stop and happy to take any questions. Thank you. They say that if you don't have any question, then everyone got everything or nobody got anything.
Thank you, Dr. Jain. Thank you for nice question, talk on nanopore on the whole. So people, we are open for question, and yeah, so the basically nanopore is revolutionary in sense that it is portable and accessible to many laboratories. So indeed, in our institute we have a nanopore sequencing facility, plus PIs also have in their cupboard, so I guess. And so and I saw many places, so that's advantage of being small, and diagnostically it's important because you don't want to sequence large chunk of DNA at the same time. So question wise, others should ask question because I am in touch with him. How to search epigenetic signals in nanopore results?
How to search for, sorry?
Epigenetic signals.
Okay. So generally what happens is when you're doing modification analysis, you're working from a reference guided analysis standpoint. So which is to say that if you're looking at a CPG, you know that given the sequence, you're looking at a CPG island. And then you ask the question, now that we know we are in this kind of a sequence context, let's go back and look at the ionic current corresponding to this region in the data and ask the question if that ionic current is more similar to a canonical cytosine bearing sequences or 5-methyl cytosine bearing sequences. And basically what this means is that you initially train a model that knows what cytosine should look like in current, ionic current space, compared to what 5-methyl cytosine should look like in ionic current space. And once you've trained those models, it's very much like speech. If you teach the speech recognition algorithm the words and then ask it a sentence the next time, it will be able to infer that based on the sentence. And then if you have a different accent, if it's been trained to detect an accent, it will be able to detect the accent as well.
Okay, so any further question? Could we see lights of nanopore in non-coding RNAs?
Yes, we do. So we have seen non-coding RNAs with the nanopore, even in poly-A RNA populations. Typically, most of the RNA sequencing happens with poly-A purified RNA because people want to study mRNAs. We've sequenced tRNAs and ribosomal RNAs. We've also looked at non-coding RNAs within the mRNA population, as well as long non-coding RNAs or link RNAs, which are of their own interest. So it can be done. And generally it becomes on the question that you're trying to ask, because there are some technicalities in how you adapt RNA for the nanopore. Most of the times, poly-A is easy because all of them have a poly-A tail. RNA sequencing, if you know the 3' end of the RNA of interest that you're trying to sequence, you can do that, which is why ribosomes and tRNAs and mRNAs are easy. The non-coding RNAs, other non-coding RNAs as link RNAs are doable, and there are techniques that one could use.
Yeah, so there is second part in the question. So how is how to investigate the expression in disease relevant regions, say all cancer or immunomodular diseases, how non-coding RNA will play a role in human disorders, human disease disorders or cancer or something.
It's a very good question, and I think you have more of people who are more of an expert at non-coding RNA biology than I am. But my understanding is that in my interest and the stuff that I've done with collaborators, it's going to be important. Some of it is we do not know yet. A lot of that, at least in our work, is hypothesis driven, where somebody goes and does something, for example, with macrophages, human or mouse, and you find a stimulant that changes some expression profiles, and then you go and try and analyze how non-coding RNAs are impacting some of the expression profiles in terms of how quantifiable the expression is within reason aside from the rare abundance transcripts, which are difficult to quantify with any technology. For what I would call the conventional expression transcripts, it's very comparable in terms of how good of a quantification correlation happens with nanopore versus other technologies.
In this regard, I have another question. Normally, what we see non-coding region, they are marked by massive repeats. Yeah, so there's ultra-conserved regions, say, or fun-seek regions. So but they are marked by massive amount of repeats, and then what happens? Say, we are in cancer lab, and then we do not want to. At some point, we are questioning ourselves whether we are going into the right direction, because you do not want to come up with a marker which next month somebody else is thrashing you out, and it do happens very often in the papers, and so people will go to the coding or which are not in the repetitive regions. But most of the ultra conserved non-coding critical stretches are in those extensively repeats regions or in between them. So can those problems can be resolved combining nanopore that you have sequencing correctly in one read, probably?
Yes, I think so. The reason for that is because of the repetitive nature, length becomes your friend, because you are reading long enough stretches, and once you find anchor points on either side of the repetitive sequence, that is a good thing. So it's kind of the same way that when we go about repetitive sequences like centromeres and telomeres, in that if you have a long enough read, then you can start anchoring it on either end, ideally both ends of the repeat. And same thing with the non-coding RNAs. The good thing, I mean, again, there's obviously some upper limitations. So for example, if we were to just conventionally try to sequence Titan, which is the longest known protein coding RNA, 100 kb is a bit of a far stretch. But in the normal range, which I would call like the 5 to 10, 20 kb range of RNA strands, that's the length becomes your friend. So I think that would be largely the case.
And then I have another question that wait, I forgot, but I'll come back to it. It was a very interesting question which came out of my mind, but I'll come back to you privately later on. But Gaurav Sharma is asking a question. I want to know your views as a comparative or comparison between nanopore and PacBio Hi-Fi.
Hi, can I?
Sure, please go ahead, Gaurav.
It was wonderful talking. I really loved it. And especially, I have read about nanopore, but this concept picture which you have shown, that was really fascinating. And I remember, when I was hearing some talk at one point of time, I realized that, Professor Deemer, he was at UC Davis. So I thought, because my postdoc is also from there, and that was wonderful to connect. Yeah, so I want to know, as an expert in nanopore, what are your views on nanopore versus PacBio Hi-Fi reads? That recently PacBio released this Hi-Fi concept, and
yes,
it is Hi-Fi. Yes, they call it Hi-Fi.
Yes, no, it is, and so two quick things. Yes, you're absolutely right. Dave used to be at UC Davis, Professor Deemer, and I think Dave came to Santa Cruz in the early 90s. He moved to Santa Cruz for a good personal reason at the time, and then he's been at UCSC ever since. The patent that I talked about is jointly owned by UC, University of California, and Harvard regions. And to your point about nanopore versus PacBio, it's an interesting question. One of the other projects that we have, that we are involved in from Santa Cruz standpoint, is the human pangenome, which is essentially to come up with the best recipe to assemble multiple human reference genomes at an affordable scale. And that becomes a technology bake-off as well. We are just finishing up an assembly bake-off as part of that, where the principal contenders for long reads are nanopore and PacBio Hi-Fi. What the Hi-Fi reads are amazing at is that you are getting on those 15 to 19 kb long reads with nearly 99 or 99.9% quality or accuracy. So basically Q20 and Q30 level reads. What that translates to is straight out of the assemblies, you get Q50 plus level assemblies that are also phased. So it's fantastic. And for most of the regions of the genome, for most of the segmental duplications, it's wonderful. Where the limitations start arising is once you start looking at the hard to map regions, which are repeat rich, could be telomeric, centromeric regions, and scaffolding across those regions, where the ultra long or the 100 kb plus long nanopore reads become very useful and applicable because it has its own virtue. And then combined with the methylation profiles that you can learn from the reads, that adds yet another facet. So I think it becomes largely dominated by the question. For standard assemblies right now, if I had to just do a standard assembly, Hi-Fi would give you a pretty damn good assembly. But if you were to do a much more complete job at the assembly, then they are more complementary than competitors.
Okay. Yeah, no, that's a valid explanation, I think. Yeah. And what about like the quality scores, like as compared to, because PacBio Hi-Fi is saying like, they are giving quality similar to Illumina HiSat. Okay, but what about nanopore? Because like, what are your views?
So 90% accuracy translates to Q10. 95% is you're looking at mid Q15-ish, and which is fairly reproducible at this point. And generally, what we do is we take some element of that now I'm talking about genome assembly side of things. When we are polishing draft assemblies, we'll do filtering based on not only mapping quality but also base quality, because you might have local regions within reads given the mapping that are at times lower quality. So you can sort of not mask them, ignore them in drawing consensus accuracies and consensus polishing approaches and so on and so forth. So in nanopore side, that's kind of where it is. We generally do not filter reads out. We can't do Q30 level filtering at the moment in the reads. But what we find is that having more reads is fine. Most of the assembly approaches we do, what we have found is that at least in our framework, we use an assembler called Shasta, which removes reads below 10 kb in the initial assembly step. And as an outcome of that, basically Shasta can do human genome assemblies in a matter of 3 hours. To give you an idea why this is so cool, is the human genome paper that we did in 2018 took 150,000 CPU hours and would cost about $20,000 on Amazon EC2 right now. The same thing assembled with Shasta would take $70 in 3 hours. So it's a very big iteration ability with that. So, and by removing the short reads, what happens is it happens to be the case that you also remove a lot of the low quality reads just as an artifact or side effect of that.
Right.
In the assembly step.
I have means because I have used PacBio, not the HiSeq one, but P64. We used to use that earlier. And so what they used to do, like, they remove the short reads earlier and then they use it for consensus polishing later on.
Yes, exactly. So most of them do that. The reason the short reads, part of the reason why they do that, and we do as well, is most of the line elements in human genomes are about 6 kb. So if you remove things below 10 kb, you reduce the complexity of the graph substantially. And once you have a graph genome, then you can come back and reuse the short reads given the mapping.
Okay. No, thank you very much. Really enjoyed your talk. Very, very great.
Yes, very kind.
So since we are going a little bit technical, so I come up with a question which I forgot. We have nanopore, PacBio, and Illumina for a non-model organism. And how do you approach actually? So you do a PacBio first, or how you'll mix it? How you'll polish actually? If you
There's a few ways. It kind of, I think, it somewhat depends on some factors that you don't know a priori. You can get a basic estimate of the genome size with counting k-mers from Illumina. I would, for example, do that, and then also do independent test assemblies with both Nanopore and PacBio individually, and see what the outcome is. How complete does the assembly look? And if you know any closely related species that you can use the annotation information for, then also look at how complete the assembly is with the BOOSCO analysis from something like POST, for example. And then you start iterating over the combinations. So Nanopore plus Illumina hybrid or PacBio plus Illumina hybrid. What changes a lot of these things is the genome size, the genome complexity, the repetitive nature in the genome, GC content, and so on and so forth. So it's a little ebb and flow architecture, but that's kind of where I would start.
Okay. So yeah, so that's really nice. And if no question, we will thank the speaker, Dr. Jain, for coming up so early in the morning, pre-breakfast, and delivering a nice lecture, and then follow up with the questions. So we will be in touch, and we will like to thank you from entire organizing team, from the parts of participants, and for entire team of Institute of Bioinformatics is thankful for you to come up for this webinar. And thanks again, and thanks to entire of your team for nice work. And we keep on following.
Happy to be here.
Yeah. So thanks again. Yeah. So with this, we are coming to an end, and then we are open for a general question, and then I will tell you that it was a nice experience because as I see that we are registration over 2200, and that was really international in a way that we have 24 country participants. So I will name that those countries because that's very important. We have participants from Azerbaijan, we have participants from Bangladesh, Brazil, Canada, China, Egypt, Ethiopia, Germany, Hungary, Israel, India. Obviously, we had a speaker from Israel as well. India, obviously, Indonesia, Japan, Kenya, Kyrgyzstan of a region of Iraq, and Kyrgyzstan is slightly on the top region, and where people have proudly say that this is that part of Iraq. So basically from Iraq, Malaysia, Nigeria, Norway, Oman, Pakistan, Portugal, South Africa, Sweden, USA, and obviously from USA we have speakers as well. So with this, I would say that this is a remarkable event in terms of participants coming from different time zone and participating. And we have decided that we will give you certificates for all registrants who have registered in the Google form, and it will be automatically generated when we sit next after the meeting for evaluation. And we will send all of you because in the pandemic now the schools are not taking an exam, so we are not counting how many hours and you participated, you registered, you participated, you benefited. Obviously the videos will be available on YouTube so that you are able to see whenever you have time, and we have to be generous on that. And so do not worry about getting your certificates. Your certificate will be in your email in next 15 days whenever we sit down and do the analysis. We do not have any feedback link, however your feedbacks are always valuable for next, so you can just message feedback and email me subject feedback, and email me to my email ID, and we will take care of your feedback. And obviously there are a lot of scope for improvement, and we have noted down. And this was my first one-man organization. So with that one man, in a way that we had everything in IT and presentation and everything I was managing, because we do not have office open, and so in that way we were managing. So but we will take account of that, and that all the videos will be on a YouTube channel, and so that you have access to that And so with this, we also like to thank today's speakers who are here. Thank you all for coming, and obviously we will be open for some questions and your suggestions. Somebody has a specific question, and those who really need the certificates, because for school or college or university, they should be registering in the that will be better. In case you do not get in next 2 weeks, you have rights to write back and ask for it. We will still be able to do it. And obviously we have a timeline, we have recorded that who were present from where and all. So no problem, you can tell me your name and we will generate it, because I like you are writing me and I am seeing that you are there. So by the time many of names by watching itself I remember it actually. So and we have method of and in such a big meeting, if 5 or 10 are benefiting by not being present, knowledge can get certificate still, but I recommend those who are not participated, please watch the video, and knowledge is more important than any certificate. So nobody is asking me which meeting I attended 10 years back, but the knowledge I acquired is very important to carry forward. So with this we come up to an end, but if you have a question, we will be taking on, and we are going off from YouTube. And we really thank everybody from local organizing Institute of Bioinformatics, and those who have participated. Some of them were not in organizing list, but they were participating and helping in the organization. So and those who also asked the questions and all. So no, a certificate will be given to everybody. So yeah, thank you. My name is Akansha. I'm just registered. Yeah, even if you are not registered, you can register still. The form will be on. Yeah, form is available on the. Anybody who has not registered can register in the form, and we will know at the time. By time will be there that you registered after that. So, and some of you have registered more than once. So we will sort minus you will do, then only you will get the certificate. So basically you get one copy, so that
Sir, when will be the links available of YouTube?
Links of YouTube's, if you follow today's link I had given, right? So you can go to the channel, and then the channel, it's available on the channel, but we will
you know what has happened, sir? Yesterday when I tried to enter the seminar, nobody let me in. It was showing, only on those windows when we enter. Yesterday.
So anybody who do not find YouTube, so this currently YouTube link I have given. This YouTube link has you can go to subscribe that channel, and the channel has the video, and channel will have more videos. In coming days we will put all our internal meetings as well, where we are having presentation.
There are two. Okay. The second one, YouTube meet. Okay, sir. All the 3 days links will be available there.
Yeah.
I mean,
So and there are a lot of perks available with that. So a lot of
because sir, now day 1 and day 2 thing, it's not showing over there. That's why I asked.
No, it's okay. That's correct. Your question is correct, and discussing is important. So thank you. Thank you.
Thanks a lot, sir. That was very nicely organized, and thank you all for your. For the
so anybody, if you have a question, you can always reach out, and we will be able to answer your queries because you are the participant. We do not charge you, but in a way we charge you because your valuable time you gave. So time is money. So and you also hopefully learned a lot, and I learned a lot from others, and so basically we value each and every participant, and we're thankful to them for joining, and we will be giving your queries proper answer. So no query will be left out. So we will be replying your response will be given. And we are since we have more than 2000 email IDs, Google server was not allowing me to send a common email. So bulk emailing was not allowed, and it's also not considered good. So but individual queries will be answered. And so with this we are really thankful, and COVID-19 webinar overall is coming to an end, and we are really thankful to everyone. And with this, I'm going out of YouTube first, and then we'll be here for some more minutes in the Google Meet, if anybody has some. So